Good morning, ladies and gentlemen. We'd like to officially welcome you to our seminar and workshop today titled as Harnessing Long-Read Sequencing for Answering Priority Health Concerns, a revolutionary seminar and workshop in genomics and precision medicine for healthcare sector. Brought to you by Genomica Indonesia in collaboration with BGSI, BB Binomica, and UNDP. My name is Ragria Gelih Nandira, and it is my absolute pleasure to be your MC today. Our event commemorates BGSI's second anniversary, and with it, we have prepared several experts across multiple sessions and workshops to celebrate the advancement of genomics field in Indonesia, spearheaded by the presence of BGSI and Genomica Indonesia. Before we begin the first session, I'd like to invite Ibu Ines Atmo Sukarto from BB Binomica to give her warm remarks to start us off today. Please join me in welcoming Ibu Ines Atmo Sukarto.
Everyone. It is great to see so many familiar faces from around Indonesia, all the various labs here, but it is also great to see some new friends that have donated some of their time to come over here and share some of their experiences. So I'd like to welcome Dr. Ira Davidson from the Garvan Institute. Ira, it's great to see you here, and thank you for making this visit to Indonesia, your first one, I believe, and I hope one that will start many more trips here. I would also like to welcome Dr. Tidapit Wongsurawat from Thailand, Jili from OND, and Rebecca Chesterfield from OND as well. So today marks the second anniversary of BGSI. Many of you are probably familiar with this national program for precision medicine that was launched in August 2022 by the Minister of Health. It started as a big, grand idea. I look at Prof. Gunadi, who has been on this journey with me and many others. It started with trying to convince the Minister of Health and the Ministry that this was a very important program. We had to also convince a lot of the hospitals to join us on the journey to build a strong ecosystem. But it's great to see that after 2 years, we have 10 of the Ministry of Health hospitals that have joined this precision medicine initiative. We have, over the 2 years, developed a two-tiered biobank, which stores now up to 8,000 samples from participants that have been recruited from all over Indonesia. The 10 hospitals focus on 9 diseases which has been selected by the Ministry of Health as diseases of great importance to Indonesia. So I think 70% of the samples in the biobank are from patients with diabetes, cardiovascular disease. We have rare disease patients as well, and we hope to see this biobanking grow over the next years. I think the aim is to have 10,000 by the end of this year, and then 18,000 samples every year for the next 5 years to try and reach 100,000 samples. Next year, the program will also start a recruitment from the general population as well. So we will not only store samples from patients and controls, so I'll have to say 30% of the samples at the moment are from controls. I was looking at the data yesterday. It appears that our oldest participant is 104 years old. That's amazing. I thought it was an entry mistake, so I rang up the hospital. It's like, are you sure you have these participants that's 104 years? And it's, yeah. The person was born in 1920. The youngest participants are, I think, a year old. They tend to be rare disease patients. So we've, the collection is quite diverse. 55%, I think, female. Average age is about 46 and a half years old. So this is an asset that is being built and that I hope will be utilized by researchers, some of whom I think are going to be in this room, so that the data that is being generated can actually underpin a lot of new research in Indonesia. the whole genome sequence data that is generated by Bebe Binomica will be accessible, will be made accessible to researchers. And I hope that in the years to come, many PhDs will come out of analyzing and looking at this data and utilizing the samples. So I think today is going to be a day of learning and of sharing. Nice to see you. I follow you on Twitter. It's great to see what Thailand has been doing, and I hope that you will be able to share this enthusiasm with the researchers in the room. I think for the participants here, I hope that you will be asking a lot of questions and actually make use of all the experts that you have in the room. If you've been having issues or problems, this is the time to try and share and discuss and try to solve problems together, because I think that's also, at the end of the day, it's what ONT is about. It's a community of scientists that are trying to use this technology. So I wish you a fruitful workshop. And I hope that from this, a lot of very interesting data analysis will come. So I guess it is my role to declare the workshop open. Thank you.
Thank you, Bu Ines, for your wonderful remarks. But now I'd like to invite Bu Ines back, and then all the speakers and then moderators. We have to take a picture to commemorate the beginning of this wonderful event today. So I'd like to invite every speakers and moderators, facilitators that we have invited today to take a brief picture. Are the photographers ready?
Recording
please,
in
get up on
progress.
the stage. Yeah. All right. Okay, thank you so much. Please return to your seat. Thank you so much. Okay. All right. Next, I'd like to invite our moderator to help us lead the upcoming sessions and the respective Q&A sessions afterwards. So please join me in introducing Dr. Mutia Ayu Putri Komaheri.
Thank you, Rakria. A very good morning, ladies and gentlemen. My name is Mutia, and it is such a pleasure to facilitate today's session. So we're now at our first session of the day. Okay, and there are a few housekeeping items before we begin. So we'll have 3 speakers for this session, and at the end of all talks, I would like to invite all speakers on stage to sit here for our Q&A session. So please ask questions during the Q&A session. For our online viewers, please ask your questions at any time during the talk in the comment section, both in YouTube or in Zoom. We also welcome questions in Bahasa Indonesia. Our team will help to translate your questions to the speakers. Kami mengundang Bapak Ibu untuk memberikan pertanyaan dalam bahasa Indonesia. Nanti tim kami akan membantu untuk menerjemahkan pertanyaan tersebut. So now into our first talk for today, it will be a joint talk by our very own dynamic duo, Dr. Ariel Pradipta and Himawan Mashihuri. So the title of their talk would be Long-Read Sequencing for Addressing Disease Mechanisms. We are honored to have with us today Dr. Ariel Pradipta, a distinguished clinician scientist. He has an MD from Indonesia and a PhD from Osaka University in Tokyo. And his research career has spanned prestigious institutions in Indonesia and internationally. And most notably, he served as a specially appointed researcher in immunopathology at the Frontier Research Center in Osaka, Japan. He currently serves as Chief Scientific Officer at Genomics Solidaritas Indonesia and holds a teaching position at the Faculty of Medicine Universitas Indonesia. His expertise lies in understanding how our bodies defend against various pathogens. Now, Himawan Masyihuri is the lead technical support team at Genomica Indonesia, and in this role, he oversees application of Oxford Nanopore technologies, as well as managing numerous sequencing projects and conducts training sessions across Indonesia. Are you okay? Yeah, okay. And his prior experience includes serving as a laboratory coordinator for COVID-19 PCR testing and genomic surveillance, where he contributed significantly to pandemic response efforts. So please join me to welcome Dr. Ariel and Himawan.
Right. Okay, so I was told I will get a pointer and a controller. So good morning, everyone. Morning. Thank you. Nope. Does the pointer work? Is there a pointer function? No? Oh, this is just for next. All right, okay. Sorry. All right, so good morning. Assalamualaikum warahmatullahi wabarakatuh. Thank you very much. My name is Dr. Ariel Pradipta. Thank you very much, Dr. Mutia, for the very warm welcome. I've been told by my students that I talk a lot, so I'm going to set a timer for myself. Because the 30 minutes is actually need to be split between myself and the distinguished Mr. Himawan over there, because we need to deliver a very interesting journey. So today I will try to display the potentials and the journey, the difficulties, and the heartfelt moments of being able to utilize long-read sequencing for investigations for disease mechanisms. Now, the challenge here is for us to define disease mechanisms. So I'm gonna tell a story that came from my own PhD journey when I was studying in Osaka University. So I was told by my sensei, Yamamoto sensei, his name was, a very unique gentleman and a very smart scientist, that there's two ways to think about immunopathology, because that was my PhD on. And the immunopathology can be thought from a thinking process that starts from structure, and then we go into the different function and the dynamics of each of those functions. Now, the other way is that each of those structures in immunology especially, and later on we will see in genomics, rarely is involved in only one dynamic. They are involved in multiple dynamics, and each of those dynamics lead to several plot twists. Now, this occurs in both host immunology and pathogen immunoresponse. This also happens if you are, I'm sure you are all very adept at reading all the different genomics and whole genome sequencing studies, genome-wide array studies, and all those different things. We will see that different dynamics can relate to several functions, especially diseases. Now, in a simplified way that totally undermined my 5 years of blood, sweat, and tears in PhD, we began by looking at two very important gene sequences, which is the IRGs and the GBPs. Now, both of them are important in recognizing intracellular pathogens. And then through the magic of CRISPR and the magic of genome editing, we created, I was told, I was assigned to create a knockout mice. So I knocked out that specific IRGM2, and I found out that the mice that doesn't have that gene structure basically led to their death after they're infected by a certain pathogen compared to wild type. So obviously, that certain gene is very important, and whatever protein that comes with it. So we had to find out what was the function. At this point, we know that the function is to keep the mice alive after an infection. But what's the dynamics behind that? To put it simply, this particular gene produces a protein that attaches to a vacuole that comes inside your cells after they're infected by a certain parasite or bacteria. And then they allow the body to decide, the innate immune system, to decide whether we should kill the parasite or that this is a host cell, that it does not need to be killed. So the gene itself, the protein that is produced by the gene itself, does not lead to the killing. It only leads to recognition on whether this is friends or foe. Now, when we think about it that way, there's a very simplified way of doing it. So after I try to mutate, I try to make different kinds of that protein with changing different sections and name them all these wonderful numbers, M77A. That's actually the name of the amino acid position and the change that I did. And I found out that by modifying those specific parts of that structure leads to different activities. One mutation doesn't allow parasite killing. The other mutation still allows killing, but it doesn't allow antigen presentation. The most severe one doesn't allow everything. It doesn't allow antigen presentation, and doesn't allow parasite killing. And all of these kinds of explanation is now trendified, being made very knowledge by this one term that everybody keeps saying, omics. It's like everything now ends with an omic. There's a transcriptomic, there's a metabolomics, and there's genomics. Now, all of this is just part of a figure, part of the journey that we want to figure out disease mechanisms. And those disease mechanisms, again, trying to remind us, comes from both analysis of upstream and downstream. Now, how we view that tends to be very different when you're a clinician or when you're a molecular biologist, when you're a microbiologist or when you're a clinical pathologist, when you're a pathology anatomist or whether you're involved in oncology. The upstream and the downstream are very, very different. But we are lucky because this is part of a big journey to realize dream to reality that started way back in 2002 and accelerated very quickly from 2004. In 2002 was the first announcement of the genome-wide array study, the GWAS. And then later on, in 2005, is the emergence of next-generation sequencing. Now, next-generation sequencing allowed us to high-throughputly to analyze the sequence of the DNA and the downstream activities in a more rapid way. Then, we need to discuss about what's the analysis and how we connect genotype to phenotype and all that, and I will tell us all about that challenge on another lecture, because I only have 7 minutes left. Right. So in this case, all of this is part of the dream to reality that is, it's kind of realized, and I wouldn't say realized, but precipitated in 2017 when it was the first case report of gene therapy for sickle cell anemia hemophilia, and the first human testing of immunotherapy against sequencing-defined patient-specific neoantigens, which is Bu Ines have pleasantly introduced to us using the terms precision medicine. Now, these are the kind of things that are accelerated by next-generation sequencing and the ability to do long-read sequencing for the completeness of the reads. Now, this is something that relates to me because I use glasses and I have myopia, and the kinds of analysis that comes from there allows us to find that there are genes, for example, sorry, this is too small, you're welcome to find the paper later, but there are genes that relates to how myopia progresses. And all of this came from next-generation sequencing. Now, to discover disease mechanisms, they actually had to sequence in animals too and find homologs of those genes in animals and in humans and try to discover, this is in animals, they make animals have glasses and then they try to cure their myopia by taking off the glasses. And they found that there are different genes that are overactivated or underactivated from a transcriptomics point of view. And from an ophthalmological insight, we saw that this is, again, part of a very long journey of detection, for example, from 1869, the first description of congenital amaurosis, which is, unfortunately, blindness. After 131 years, through the development of all these things, there's a successful gene therapy for Leber's congenital amaurosis in dogs. All right? So, but now the clinical trial is open, and I think you can check it out in clinicaltrials.gov. Now, all of that is part of the journey of how we had to basically find out about these disease mechanisms. And turning science fiction into science is made possible because there is groundbreaking treatments that are allowed right now, that are enabled to the many different analyses that we can do. Now, delivering these discoveries is a whole different story. And to describe this without taking the thunder out of our next wonderful speakers, I will talk about one that relates close to immunology, which is methylation and epigenetics. Now, a lot of methods have been done to analyze epigenetics and methylation, but one of the most versatile ones is using long read because it allows for non-modified analysis. Now, there are there's two very important journeys that I would like to introduce today. The first one is from the structural molecular biology, in which they found, through methylation analysis, a section, the green one over there, the unique section, that is very important in nucleosome remodeling. That is a very structural approach. It is not about phenotype that comes later, but it is a structural genetic analysis. Now, it actually relates to aging, because one of the most important mechanisms of aging is genomic instability that is aggravated or alleviated based on their epigenetic alterations. So these are the kinds of disease mechanisms, and I guess in terms of aging, it's not much of a disease. But as again, apparently we found a 104 years old just now that was mentioned by Buines. I swear I didn't plan this, Buines, but apparently it becomes very relevant because these are the kinds of things that we can discover right now. On the other side of it, from a diagnostic point of view, one of the most important findings in terms of using long read is the diagnostics for meningioma. Because WHO grading now have adopted the use of methylation status for them to grade the meningiomas. And these are the different kinds of analysis that are needed to complete this grading based on the new WHO staging. I think there's some attendance from PON, and I think I cooperate mostly with Dr. Yuli and Dr. Cynthia. I hope they're here and very excited to see this. And with a lot of this development leads to a very conducive environment to talk about the use of epigenetics analysis for discovering disease mechanisms. Now, the downstream of that, or maybe upstream of it, again, depends on how you look at it, is the different ways that we can analyze transcripts. Because we want to know not just from the gene itself, but also the expression and the protein existence later on. There are several steps in the middle that I simplify, but I simply do not have the credits to talk about it today. And so in terms of transcriptomics, this is an interesting one because NGS transcriptomics, next generation sequencing for transcriptomics, allows for high throughput analysis on the many different mRNAs and RNAs that needs to be analyzed through a certain sample, for example. Now, of course, there are strengths and weaknesses, and all of this require discussion. And a lot of this can be seen on the versatility of it from a publication in Nature, I think around 3 years ago, 2022, that showed the robustness of analysis for transcriptomics using next generation long-read sequencing. Now, this dynamics of progress can only be enabled if we are able to look at it I use a very basic concept that I think we're all very aware of, host, environment, and pathogen. So I want to see that the host is the lab solutions that we all have right now, that we're all excited to develop. And then the pathogen is the vision, while the environment is the ecosystem that we're building right now in this room. So the lab solutions include so many different things to be able to use it, to understand it, and to analyze and to synthesize all those different explanations. And then the ecosystem is literally the discussions that we have right now, the different collaborative efforts that hopefully can create something that is greater than the sum of its parts. And the target is to understand the different diseases that I'm sure later Dr. Tip will talk about applications in the medical school, and then Prof. Gunadi will talk about applications in rare disease, and I think Dr. Ira also will talk about that. And hopefully we will be able to lead to collaborative genomics, where we publish all these different journals and being able to collaborate with the different people around. And just on time. Because to reach that stage of collaborative genomics, as you can see, we have that lab environment, the ecosystem itself, and the ways that we have to build this ecosystem. And because of the many different diseases that we're involved right now, ranging from cancer, rare diseases, ophthalmological diseases, even infectious disease and immunology, it requires an end-to-end discussion. A place to be able to discuss about various solutions that we need to ask and answer and discuss. And with that, I would like to invite Himawan to be able to lead us in this discussion, because he will talk about how Genomica will be able to provide these solutions for you. Please, Himawan. Thank you.
Thank you so much, Dr. Adil. Okay, Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. My name is Himawan Masyuri. I'm from Genomica, Indonesia. Continuing Dr. Ali's presentation, today I'm excited to share with you how we can enhance the value of research through a multiomics approach. By integrating different layers of biological data, we can unlock deeper insight and drive more impactful discoveries in life of science. From an academic researcher perspective, using cutting-edge technologies like nanopore sequencing offer several exciting opportunities. First, it enables us to be the first to uncover novel disease biology. When working with complex diseases or unknown pathogens, having the ability to sequence real-time provides unparalleled insight into how these biological entities function and evolve. This puts researchers at the forefront of discovery, where they can contribute new findings to the scientific community. And the second, we get ultra-rich data from a single platform, which magnifies the power of the discovery. Nanopore long-read sequencing, which allows for greater resolution and clarity when analyzing genetic material. This means we can detect more significant structural variants, resolving complex genomic regions, and get comprehensive view of the biological landscape we are studying. The power of the data combined with the flexibility of the platforms allow researchers to make transformative discoveries, whether it's identifying new target for drug development or understanding the mechanism of, behind the resistance. The knowledge gain can drive major advancement in disease treatment and prevention. Okay. Ultra-rich data, From a clinician scientist perspective, nanopore sequencing offers several significant advantages in patient care. First, It plays crucial role in accelerating disease identification and diagnosis. With real-time sequencing, clinician can identify pathogen or genetic anomalies faster, which can make a critical difference in time-sensitive cases, improving overall patient care and outcomes. The platform provides ultra-rich data, allowing for a deep, comprehensive understanding of the complexity behind many diseases. This includes insight into genetic variation, mutation, and the overall genomic landscape of diseases, which might be challenging to detect using traditional methods. This enables fast treatment decision and support personalized medicine. By using the genomic data from patients, clinicians can tailor treatment more accurately, ensuring that each patient receives the most effective therapy based on their individual genetic profile. This personalized approach not only improves treatment efficacy, but also enhances long-term patient care. In this particular case, the method was applied to a family where a child was suspected of having a pathogenic variant linked to acrodermatitis enteropathica. Traditional testing method can be time-consuming, but with this 3-hour sequencing workflow, clinicians were able to quickly isolate DNA, prepare libraries, sequencing the genome, and obtain actionable results. The same approach can be applied to other patients, especially when there is a high suspicion of genetic disorder. This allows for faster, more accurate diagnosis, which can guide treatment decision more efficiently. Clinical testing for imprinting disorders such as Prader-Willi and Angelman syndromes traditionally involves multiple and sequential tests. This process is time-consuming, costly, and sometimes inconclusive, especially when multiple tests are required to identify variant and methylation patterns. Long-read sequencing facilitates comprehensive evaluation of copy number variant or CNV, single nucleotides variant or SNV, structural variant or SV, and methylation patterns. Methylation analysis provides detailed methylation pattern data at key CpG sites specific to imprinting disorder, accurately in identifying disease-specific methylation changes. Real-time sequencing offers rapid turnaround times, which increase diagnostic efficiency, and reducing the time to diagnosis. Reduce the need for multiple and sequential tests, lowering overall testing costs and simplifying the testing gaps. Clinical labs are also likely get benefit from this transition, as a reduction in test complexity and overall number of tests offered will reduce cost and lower administrative burden. Together, the shift of standard clinical testing from a stepwise system to integrated approach will have broad benefit, lead to novel biological insight, and improve outcomes for individuals and their families with rare genetic conditions. Okay. Let's talk about why multiomics is crucial for tissue research. Understanding these complex diseases like cancer or neurological disorder requires looking for changes across multiple molecular levels. And multiomics really helps to pinpoint how those genetic mutation, epigenetic changes, and shift in gene all works together to drive diseases. This approach is valuable to discovering biomarker by examining a range of molecular data. We can find more reliable marker for diagnosing diseases and predicting how the patient will respond to treatment. It can also help identify new therapeutic target by highlighting which gene, which protein is abnormal in disease tissue. All of this leads to personalized medicine. Multiomics allows us to stratify patients based on their unique molecular profile, so we can tailor treatment to each individual. Therefore, multiomics can provide a powerful and comprehensive toolset for tackling the complexities of tissue and diseases. It's transforming how we approach research and treatment, leading to a better diagnostic, more effective therapies, and personalized care. Here is the example how Nanopore Multomics simplifies genomic and epigenomic analysis with our DNA sequencing kit, allowing researchers to reveal more biology with less to get the same multiomics information with a single Nanopore DNA library. You would need 3 different library prep run on different sequencing platform and all with separate data analysis. This would take much more time and money. But Nanopore Multomics sequencing simplifies genomic and epigenomic analysis through direct sequencing of native DNA methylation analysis that can be activated by single mouse click, enabling epigenomic information to be provided alongside truly comprehensive genomic data, including structural variants, complex variants, phasing, which are only available through long reads. First, PromethION device that we will talk about are the PromethION 2 Solo or P2 Solo to the left, and PromethION 2 Integrated or P2 Integrated as shown to the right. Both of these sequencing instruments are capable of sequencing up to 2 PromethION flow cells at the same time. The P2 Solo uses the computational resources from GridION or a computer or laptop to perform high-yield sequencing and compatible with Windows, Linux, and Mac OS operating system. On the other hand, the P2 Integrated is all-in-one sequencing device equipped with an integrated NVIDIA Ampere GPU and built-in touchscreen for easy control of the sequencing run. To get started, all it requires is a power sources and internet connection. The device comes preloaded with the Ubuntu operating system and Mino software. The P2 Integrated features a more powerful onboard GPU compared to the GridION. Choosing the P2 Integrated over the P2 Solo and GridION combo will provide the advantage of faster base calling speed. The PromethION is the largest of our sequencing device, available in 2 formats. The PromethION 24 that can run up to 24 flow cells, and PromethION 48 that can run up to 48 flow cells. In both cases, a flow cell can be run simultaneously or individually. Every flow cell may deliver in excess of 150 gigabases of DNA sequencing data, making the PromethION capable of generating many terabases in a single run. PromethION is therefore highly suited to large genome sequencing at scale, whole transcriptome experiments, population studies, including detection of small variants like SNP, indel, large variants, epigenetics, or even dark genome which are only visible through native DNA or RNA sequencing. Base calling PromethION includes a high specification compute unit with base calling accelerator capable of keeping up with the large amount of data produced by the device. Thus, introducing Nanopore Multomics Toolkits, a single platform, our PromethION for ultra-rich data from a single workflow. And the best part is achieving this comprehensive genomic, epigenomic, small and large variants, and transcriptomic insight, including gene expression, splice variants, even single cell, doesn't require a new workflow. Researchers can use our exciting direct RNA and DNA sequencing. We can seamlessly integrate multiomics into their current research process without the need any additional complex protocol, chemical modification, and etc. With Nanopore sequencing, scientists can now uncover many new layers of biological information, leading to a more complete genomics picture of disease mechanism. This holistic approach not only advances our scientific knowledge, but also paves the way for new diagnostic and therapeutic. Genomica Indonesia is your gateway to advanced genomic sequencing solution in Indonesia. We offer a comprehensive range of products and services for genomic research, education, and clinical testing. We are proud to be a leading initiator to develop in Indonesia and representing the top genomic solution around the world. We are committed to achieving both national and international accreditation and ensuring full compliance. One solution for your genomic research, Genomica Indonesia. I'm Himawan Masyuri from Genomica Indonesia. Thank you.
Thank you, Dr. Ariel and Himawan. And now we're on to our second talk for today. I would like to welcome Dr. Ira Diefsen. Dr. Ira Diefsen is the bio, please. Yeah. Dr. Ira Diefsen is the laboratory head at the Genomic Technologies Group and the team leader for production long-read sequencing at the Garvan Institute of Medical Research in Sydney, Australia. So he's joining us today from Australia. He also serves as a conjoint lecturer at the University of New South Wales and holds a PhD in biological science. With over 67 publications and more than 25 million Australian dollars in grant funding, his work in genomic technologies and large-scale analysis has garnered numerous awards, including the Garvan Rising Star Award. And as mentioned, it's his first time in Indonesia and in Jakarta, so let us give him a big warm welcome to the stage.
Excellent, thank you very much. Thanks for the introduction, Tia. It's a real pleasure to be here. It's very exciting to visit Indonesia for the first time. I'm a little bit sad that I didn't make it here quite in time to watch the Australia versus Indonesia football match last night, which was apparently very exciting game and a good result for both teams, I guess. So, yeah, I want to start by introducing my wonderful team, the Genomic Technologies Lab from the Garvan Institute in Sydney, including Tia, who's actually a PhD student in my team. So it's a really great team that brings together a diverse set of expertise in molecular biology, genomics, computer science, and sort of data analysis. We work on a really broad range of topics, but long-read sequencing technologies are pretty central to everything that we do. And in particular, nanopore sequencing, which is obviously the topic of today's meeting, is something we've been working on for a long time now, and sort of methods development in the nanopore sequencing space, software engineering in the nanopore sequencing space, and applications of nanopore technology and for precision medicine is really a key focus of the group. Now, it's not just because nanopore is a cool technology that's kind of fun to play with. That's not just the reason we work with it. Really, the reason we are so interested in nanopore sequencing is because we believe it's a technology that has the potential to really transform and accelerate the way in which we diagnose inherited disease. And I guess this goal, building the tools and capabilities to make that a reality, is a key motivation for everything we work on. Now, given the topic of today's meeting, I don't need to introduce the mechanism of nanopore sequencing in great detail, I don't think, but I think suffice to say, it works by transiting a DNA strand through this protein pore that's embedded in a charged membrane, and that generates the electrical current data that we can translate into DNA sequencing information in real time. But the talk that I'm going to present today focuses on a particular unique capability. It's quite nifty capability of nanopore sequencing, which is called read until, or adaptive sampling. And this is basically a trick that allows you to program a nanopore device to recognize a given DNA strand as it is entering and moving through that pore. And then based on its sequence, you can sort of dynamically reverse the charge on that pore to spit out the DNA strand if it's not something that you're interested in. And what that means is that, in theory, we can now perform targeted sequencing in a fully programmable manner. So all of the target selection of which genes you're interested in happening right there on the device without any additional molecular methodologies to select for your target sequences that you're interested in. So when this was first sort of announced by Nanopore, we thought it seemed really like a pretty cool application, and it's easy to imagine how you could use this for many different areas, particularly in clinical diagnostics. But we wanted to know whether this sort of really works, because sometimes these things are a little exaggerated, and so we sort of set out to test this capability, and we wanted a specific application to sort of test this on and see whether it worked. And the first example that we chose centers around these specific types of sequences called repeats and repeat expansion disorders. So by way of background, the human genome contains lots of these so-called short tandem repeats, or STRs, which I'll use throughout the talk. And this is essentially just a short DNA sequence motif that's repeated consecutively again and again. So CAT, 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 all in succession. And these are quite common. They make up around 7% of the human genome, and they're highly polymorphic, which means they vary in size between different individuals. So we might have one individual who has, say, 8 copies of this motif, another individual with 11 copies, and so on. But they're of particular interest because beyond that normal genetic variation between individuals, these repeats can become greatly expanded in particular individuals, and these expanded repeats are known to cause disease. They're the known pathogenic variant in many different diseases. It's quite a complicated story. These repeats are associated with many different diseases. For whatever reason, a lot of these are neurological disorders, and this includes some quite well-known examples such as Huntington's disease, Fragile X syndrome, motor neuron disease, and many more. In fact, by our count, there are at least 40 known neurogenetic disorders that are caused by repeat expansions in at least 37 different genes, which are shown in this table. And actually, this is now quite out of date since 2021. There's been many new repeat expansions described, and so this list is sort of growing all the time. Now, because these are long, highly repetitive, often very GC-rich sequences, STRs are very difficult, sometimes impossible, to analyze using short-read next-generation sequencing, so from Illumina or BGI, the sort of more established technologies. And this means that the diagnosis of these repeat expansion disorders still relies on some pretty old-fashioned hands-on molecular biology techniques, such as repeat PCRs, Southern blots, and some other techniques. Now, these are really good techniques. They work well, but they're quite hands-on, they're quite slow, they're quite labor-intensive, and what's more, each test only tests for a single gene, so it's like a one gene, one test kind of paradigm. And that's a bit of a problem because we have, at least 40 different genes here that are potentially in play. So a one gene, one test paradigm is not going to work very effectively for this particular space. So we thought this was a really good case for us to use our Nanopore sequencing and deploy this read until or adaptive sampling functionality to programmatically target every single one of these repeat expansion genes in a single test, a single sequencing assay. So we basically designed a read until or adaptive sampling panel that has all of our STR genes, and we use this to perform targeted sequencing on the handheld MinION sequencing device. And here I'm showing you basically the sequencing coverage across a single gene, HTT, which is the gene that causes Huntington's disease. And by comparison to the sort of off-target regions, which are shown in red, you can see we get this nice enrichment of sequencing coverage across the body of the gene. So this is working really well. We're sequencing more of the gene and less of the rest of the genome that we're not interested in. And I just sort of mentioned that this work was done a couple of years ago, and we were using the sort of handheld MinION device. Now we're using the slightly larger P2 Solo device, which you guys just saw in the last talk. And this makes it possible for us to sequence, say, up to 4 patients on a single flow cell using this adaptive sequencing approach. So this looks really good. But what we're actually interested in is obviously our repeat sequence. And so at a specific position within that gene, HTT, there is a CAG repeat that has around 20 copies in healthy individuals. And so if we get sort of 30 to 40-fold sequencing coverage depth on that gene, we might get about 30 reads that fully span that repeat. And we can sort of phase these reads out into the maternal and paternal alleles, and then we can assemble a consensus repeat sequence for each allele. And so you can see for this patient, we've assembled one allele that has 64 copies of this CAG repeat, which is well into what is considered the pathogenic size range for this gene. So unfortunately, this would be a positive diagnosis for Huntington's disease. So that was kind of one patient. But in the next slide, we've genotyped the HTT repeat sequence across 12 different individuals, and 5 of which are known to have Huntington's disease. So you can see that this repeat sequence that we've assembled is very diverse. We get lots of different alleles. Every individual essentially has a unique pair of sequence alleles here, and they're covering a range of different sizes. But you can see that the 5 individuals who have Huntington's disease have a very clearly expanded repeats that are all longer than that known pathogenic cutoff for this disease, whereas our 7 individuals who don't have Huntington's all have shorter repeats that are shorter than that pathogenic cutoff. And so in this example, our test is very clearly and correctly distinguishing the normal from affected individuals, which is exactly what the test is designed to do. And when we sort of compare our assay to the standard molecular testing approaches that I mentioned, these sort of old-fashioned one gene, one test methods, across many different genes and many different patients, the results are highly concordant, which is to say that when we measure the size of the repeat with our Nanopore assay, we're getting a very similar size to what you would get with the established diagnostic technologies. Now, for Huntington's disease, this is good, but it's maybe not necessarily a game changer. But for some of these other disorders that have more complex repeats than that simple CAG example, we really start to see the advantages of this long-read sequencing approach. So I'll quickly touch on 2 of these disorders next, Fragile X syndrome and Canvas, to give you a taste of what I mean. So in the case of Fragile X, the Nanopore data allows us not just to measure the repeat and the size of that repeat, but also to determine the DNA methylation profile of the FMR1 gene, which is where this repeat occurs. And better yet, we can do this in an allele-specific fashion. So in this subject here, we see that the expanded STR allele, unlike the normal STR allele, is hypermethylated and silenced. And this epigenetic silencing mechanism is actually central to the pathogenicity of this repeat expansion in Fragile X syndrome. And so being able to identify this silencing mechanism in addition to the repeat size and in an allele-specific fashion in a single assay without using multiple different techniques is really a key advantage of this technology. The next disorder that I'm going to talk about is an ataxia known as Canvas. So this is a movement disorder that's characterized by loss of muscle control that can have quite devastating consequences for the patient. And this is caused by an STR expansion in a gene RFC1. So RFC1 expansions are relatively common among the normal population, and they can be very, very large, sometimes around 5 kilobases in length. And they come in basically a bunch of different flavors. So it's a 5 base pair repeat, a pentanucleotide. In healthy people, you generally see either one of these 2 repeat motifs that are shown here in pink or orange, which are considered to be non-pathogenic regardless of how large they are. So they don't cause disease if they're pink and orange motif. Then the main pathogenic repeat motif is this 3rd, more rare sequence motif shown in blue. And if you get 2 copies of this pathogenic sequence, that results in Canvas, so it causes disease. And so for the genetic diagnosis of Canvas, we need to know several different pieces of information. We need to know, is there an expansion present in RFC1? How large is the expansion? We need to know whether the expansion is on one allele or on both alleles. And we need to know what is the repeat motif. And again, we need to know that on both alleles. And so to answer all of those questions reliably, you really need multiple different tests, and sometimes it's not even possible because this is a very challenging and large sequence. And so The question was whether we could do this with our read until assay, and I'll start by showing two subjects. So these plots here show the density of each nucleotide as well as each of our different motifs that was observed across the length of this STR site. So in the subject up on the top, who's healthy, we've detected 2 copies of that pink motif, both of which are around 600 base pairs in length. In the subject underneath, who has canvas, we've detected 2 copies of the blue motif, which is that pathogenic motif, one of which is around 2.5 kb, another which is around 3.5 kb. And so this would basically be a positive diagnosis for canvas. And you can see it's quite concordant with the southern blot test over to the right, which is the sort of established molecular method, where we see kind of 2 sequencing bands corresponding to those 2 different expanded alleles. But now I bring in like a third subject who also has canvas. And this person has 2 large expansions, both of which are around 5 kilobases in size. And they show up as a single band on this person's southern blot over to the right. This person had had a separate PCR test which confirmed the presence of the blue motif. And so this basically looks like they have 2 copies of that expanded blue pathogenic sequence, which would be a positive diagnosis for canvas. However, when we sequenced this using our Nanopore sequencing and we phased out the reads, we actually found that one allele over to the right is not the pathogenic sequence, but corresponds to the orange motif, which is currently considered non-pathogenic, so non-disease causing. But this is really quite interesting because this person has canvas. So by the conventional wisdom, they shouldn't have canvas because they don't have 2 copies of the blue sequence. And so what that tells us is that most likely this orange motif, if it's expanded beyond a certain size, can in fact be pathogenic. It can cause disease. And I guess that our assumptions that it's always non-pathogenic may have been misguided by some ambiguity in this molecular testing, which as I showed here, the old school molecular testing couldn't reveal the full picture in this case. And so I think that's a really nice, clear example of how long-read sequencing can enhance the resolution and improve the genetic diagnosis of these really complex repeat disorders. So we published that work as kind of a proof of concept 2 years ago, and since then we've been actually trying to use this method to improve the diagnosis of patients with neurogenetic disease. And essentially we're trying to demonstrate how this can be applied clinically. So one area we've been working on is for the diagnosis of spastic ataxia spectrum disorders. Canvas is one of these, but this is a group of inherited neurological diseases which manifest in progressive loss of muscle control, and this is really devastating for patients, as I said. So spastic ataxias affect around 1 in every 10,000 people, and they're quite heterogeneous. And in terms of the symptoms and the age of onset of those symptoms. And we know that there are over 450 different genes involved in this spectrum of disorders, as well as all different classes of genetic variation, ranging from single nucleotide variants, small indels, larger structural rearrangements or SVs, copy number variation or CNVs, and of course STR expansions, many of which are implicated in spastic ataxia. So as a result of this, in Australia, patients who are affected by spastic ataxia disorders, they often have to undergo many different sequential genetic tests. So they have different tests that assess different genes or assess different kinds of variation. So for example, a patient might have whole exome sequencing to identify sequence variants, followed by repeat primed PCR on a specific gene to look for a possible STR expansion. Then they might have another technique known as MLPA to look for structural variation, large deletions or copy number changes. And this process is quite slow and costly. More importantly, this kind of fragmented testing landscape means that many diagnoses are potentially missed because the right test wasn't necessarily ordered for the right patient at the right time, or perhaps the right test wasn't even available to a particular patient. And so we set out to try and, I guess, change this by designing a targeted nanopore sequencing assay that would capture all 469 genes that are currently known to be implicated in spastic ataxia, and leverage the capabilities of the long-read sequencing to simultaneously detect every class of genetic variation across every one of these genes, all in a comprehensive integrated assay. Now, to evaluate this kind of single test approach, we've so far applied this new method to a cohort of 34 genetically undiagnosed patients. So these are patients who have spastic ataxia in a phenotypic sense, but have no previous genetic diagnosis identified. And to summarize very briefly, this has worked really quite well so far. Our single test approach has so far produced a new genetic diagnosis in 41% of these patients. So this is in patients who've had no previous genetic testing. But more strikingly, in those patients who had already been through standard genetic testing pathways in Australia, but remained undiagnosed, we've found a new diagnosis in 39% of these patients. So this is a really impressive uplift in the diagnostic yield, and that's a very encouraging result. There were kind of several explanations for these new diagnoses. So in some patients, we detected an STR expansion for which there was no current test available in Australia, specifically for these genes FGF14 and RFC1, one of which I mentioned a second ago, or perhaps where a test was available but it was not ordered because it was not considered to be a likely cause for that specific patient. Outside of STR expansions, we found small sequence variants in genes who had not been considered during previous testing, and we also found one individual who had a compound heterozygous variant that could not be detected and phased using short-read exome sequencing. So one particular gene that was very important here was this gene FGF14, which contains an STR expansion with a GAA motif, which when expanded beyond 250 copies, causes a recently described ataxia known as SCAR27B. So there's no current diagnostic test available for this in Australia. And so in our cohort of 34 undiagnosed patients, we actually identified 7 patients who carried a GAA expansion in FGF14 that is sufficient for a diagnosis of SCAR27B. And this was in fact the most common genetic diagnosis made across our cohort. It's important to note here that we also picked up 6 other patients who have expanded repeats in this gene that I'm showing here without the asterisks. But these repeat expansions either fall just short of that 250 base pair cutoff, or perhaps those repeats are not pure GAA, but they have other sequence motifs mixed in there, and those are currently not considered to cause disease. So once again, it's the ability of long-read sequencing to sort of fully resolve what's going on with this repeat. It gives us both the size, but also the sequence right across that repeat, even when there's mixed multiple motifs involved. So this is a really important advantage of this technology. What's also important to point out here is that there is now a drug for aminopyridine, which is also now recommended for patients with confirmed SCAR27B. So as a result of our analysis, all 7 of these patients where we found a GAA expansion of a certain size are now being treated with this drug. So it has real immediate clinical applications and advantages for patients and their families. To dig into another example here, we found a single patient who had 2 heterozygous variants in this gene SPG7. Now both of these variants are detectable using short-read exome sequencing, and this patient had short-read exome sequencing. However, these are both recessive variants, and therefore in order for them to be considered causative of this patient's disease, we need to show that they're on opposite haplotypes. So essentially we need to show that the patient has inherited one variant from their mother and one variant from their father. Now usually this would mean we would have to sequence the parents to confirm that. However, for late onset conditions like this, the availability of both parents for sequencing is often a barrier to resolving cases like this. But with our long-read sequencing, Because we get long reads that sort of fully span this gene, we can phase this gene, and we can show that the two variants occur on opposite haplotypes. They're never on the same read. They're either on the blue reads or the pink reads, which means that we can show this patient has inherited one from their mother and one from their father without needing parental sequencing data. So this is, really streamlines this process for us and is a really, low-hanging fruit in terms of applications for long-read sequencing, is just being able to resolve cases like this, which we otherwise just couldn't do with short reads. So overall, we achieved new diagnoses in a diversity of different ways. And I guess the take-home message here is really that this approach works well. This ability to capture all of the potential genetic causes of spastic ataxia in a single assay, which is a unique capability, I think, of long-read sequencing, clearly has capacity to streamline and improve patient diagnosis. So the technology works really nicely for spastic ataxia, but before I finish up, I'd like to quickly point out that this same strategy can be applied to any genetic disease or group of diseases, and there are many difficult genes and disorders out there where we think long reads are going to be diagnostically useful. So we're currently working on lots of different disorders, trying to develop sort of streamlined tests in the way that we've approached ataxia. So we're currently working on, I've gone too far. So we're currently working on thalassemia, which is a relatively common genetic blood disorder caused by deleterious variants in the globin gene family. Now, because this includes numerous closely related genes and pseudogenes, and because structural variation in this locus is often involved, thalassemia is quite tricky to comprehensively characterize using short-read sequencing. And you can see here we get many gaps in the coverage on the short-read sequencing track, where we can't uniquely align reads to the locus, and so we get gaps in our coverage, and that's because of that homology between the different globin genes. But by contrast, we get nice uniform sequencing coverage when we use our long-read sequencing to target these regions. And it's basically the same story for this second example, which is the gene PKD1, which causes polycystic kidney disease. This is another relatively common inherited disease. It's a kidney disease, and this genetic diagnosis of this is unfortunately hampered by the fact that there are 6 pseudogenes that have almost identical sequence to PKD1. They're 99% similar, and so again, when we try to align short reads to this gene, we get a lot of gaps in the coverage where we can't uniquely align the reads. But by contrast, we get very nice even coverage across this locus using our long-read sequencing. I was going to show one more example at the end, but it's essentially the same story. Another very nasty kidney gene that we work on called MUC1 that you essentially can't sequence with short reads. But using our long-read sequencing technology, we're able to span through this very repetitive, difficult gene and ultimately build new diagnostic tools for this disease. I'm going to skip over that one, given the time. But yeah, I'm going to just finish up by thanking everyone involved, my amazing team back in Sydney, the Genomic Technologies Lab, who are a wonderful group to work with, great diversity of skills and interests, and all of our collaborators. I mean, there's too many to show, but I've listed some of the important ones here, and yeah, very, very happy to take questions in question time. Thanks everyone for listening.
And I'd also like to invite Dr. Aryan Himawan back onto the stage for us to have our Q&A session. All right. So with that, I'd like to open for the first 3 questions. If you put your hands up, someone will, yes. Gunadi first, second, third. Pak Ahmad Tomo. Okay. Yeah. Please, Prof. Gunadi.
Thank you very much, Dr. Mutia. I have actually challenged question because we have now developing ONT platform in our institution to Dr. Aira. So thank you very much for your very wonderful finding. Yeah, because I think we still have long journey to achieve such wonderful finding. So my simple question: how did you design whether you jump to directly whole genome sequencing or adaptive sampling for such structural variant which already reported previously, or just simply you just running long read whole genome sequencing? And then I wonder, I guess you use adaptive sampling for ataxia, but I wonder the number of genes is more than 200 or 300, is almost 500, because I think we have difficulty if we use more than 200 and 300, then how do you solve such more than 200 or 300 gene for adaptive sampling? Thank you.
Yeah. Thank you for the question. Thanks for the question. Yeah, so it's always a tough choice as to whether it's more appropriate to use whole genome sequencing or the adaptive sampling. We really just think for a particular case whether there is a clearly defined set of genes that are needed to provide the diagnosis. So in the case of the ataxias, we have a list of about 15 repeats and these 400 other genes, which are all of the genes known to be implicated in ataxia. If we find a variant in a gene outside of that list, it's not going to be informative at this stage. It might be useful in a research context for gene discovery, but it's not going to be diagnostically applicable. And so for this particular application, where we have a clearly defined list like that, we would always prefer adaptive sampling, just because it enables us to reduce the sequencing cost per patient by around four times. But we have many other cases where we have families with rare inherited disease that is not of a clearly defined disorder or phenotype, or where the genes linked to a specific phenotype are not well known. And in these cases, we always take a whole genome sequencing approach, because simply we don't know what we're looking for, and it requires genome-wide discovery. So you kind of have to make this decision on a case-by-case basis. But we do think the adaptive sampling approach is very effective if you know roughly what you're looking for. As to the second question of sort of how many genes, I think this is approaching the upper limit. I don't think we can do more than 500 genes. I think when your list is that long, you may as well do whole genome sequencing. I also think in this case, the gene list is growing all the time. And so one limitation of the adaptive sampling is that we can't update our analysis to include new genes in the future. We would have to resequence the patient. Whereas if we do whole genome sequencing, we can always update the analysis in the future. So that's a trade-off. And I think anything beyond 450 genes, we will switch to whole genome.
All right, so that's a great tips. Go ahead, Dr. Tip.
Hi, so my name is Tip. Thank you so much for very good talk. I have a question about the DNA extraction method for long read. So in this case, you use robot or you do manually, like ultra long read DNA extraction?
Yeah, so at this point, all of our DNA extractions are performed manually, and we use it, but it's a relatively straightforward extraction process these days. It used to be quite difficult, but now there's very good kits for high molecular weight DNA extraction, assuming the sample that you're starting with has been sort of collected and stored in the appropriate fashion. It's quite straightforward to get good high quality DNA. We haven't yet automated this process, but there are automated protocols available now for the extraction kit that we use, and so that's something we'll be hopefully implementing in the future.
I see. Thank you so much. And do you need to use G-tube to do sharing to get like a 15,000 base, something like that?
Yeah, so it is important to shear the DNA to a pretty specific size range. We sort of aim for around 20 to 25 kb. I mean, anything above 10 kb will work quite well, but you don't want very long DNA in there, or it kind of blocks up the pores and can reduce the outputs. So yeah, we use an instrument called a megaruptor to do this shearing, but G-tubes also work.
Okay.
Yep.
Thank you so much.
Okay, now on to the third question. Multomics.
Yeah, thank you, Ira. It's very interesting about the Huntington disease, because it is the anticipation where somebody actually can expect to get the disease when they get expansion of the repeats. My question is, the samples, it's coming from the blood or from other sources, or can you take it from a buccal swab, because which is much more less invasive? And have you seen this evidence, this anticipation, and how many years it takes to have this evidence of expansion? And the second issue, but more technical issues, say a laboratory wants to have some, maybe a couple of genes only, which is more effective, running adaptive sampling, because it requires at least about 0.1% of the genome, or just doing a PCR. Because when you do PCR, then you have some artifactual PCR issues. I mean, from experience, which one would you recommend? Just in case only, maybe say up to 5 genes only. Thanks.
Yeah, cool. I'll answer the one about the PCR first, I guess. If you have just a single gene, or maybe just a small list of genes, and they're quite small, and you can amplify them by PCR, I think this is the best, by far the cheapest approach. You can potentially multiplex 100 patients on one flow cell if you're just sequencing the PCR amplicon. So it reduces the cost of the test to very, very low, by comparison to adaptive sampling, where we can do, say, 4 patients per flow cell, which is cheaper than whole genome, but still not super cheap. The problem is, for a lot of these repeats, you can't actually amplify them, or when you do, you create a lot of errors, like stuttering in the length of the sequences. But some of them are just because they're highly GC rich, you can't actually amplify them at all, and so it won't work. The real advantage of the adaptive sampling is you're sequencing the native DNA. There's no molecular processes, and so what you read is exactly what was the starting material looked like. And so that's a key advantage. But for other genes, if they're not repetitive, if you just wanted to phase some variants, like the example of that SPG7 that I showed, you could just amplify that in a single amplicon and sequence it quite effectively. The question about anticipation and the sample material, all of the data that I showed is from blood, whole blood, which we find is the most convenient material to work with. However, we have also been exploring using saliva, which works pretty well. It's early days. Like, getting good quality high molecular weight DNA from saliva is not as easy as blood, but it does seem feasible. And we've had pretty good success recently sequencing saliva samples on both Oxford Nanopore and PacBio long-read sequencing. So that seems really promising. It has some limitations. There's often a lot of non-human DNA in there, which can be a problem. But it seems very good. We have also, in some of these experiments, sequenced repeats from post-mortem brain samples, where you actually see kind of a lot of somatic variation in the size of the repeats. But yeah, in general, whole blood, if possible. Hopefully in the future, saliva will make this a lot more practical.
Thank you, Ira. So that's lots of practical tips on shearing, sample source, and how to approach disease and genes with targeted or adaptive sampling. So before I move on to another set of 3 questions, I'd like to invite our viewers online to also ask questions in the comment sections, and we'll read them through if there's any that comes through. All right, so we are now open for the next 3 questions. please raise your hand so we can come and get the microphone to you. Nothing yet? Okay, do you have questions online? Not yet? All right, so my turn to ask questions then. So my first questions would be to Dr. Ariel. So you've talked a lot about the conventional DNA sequencing, but more to the unconventional applications of DNA sequencing. How, from your personal experience, how would you see that pan through?
Right, thank you. So, thank you very much, Dr. Mutian. Thank you very much, Dr. Ira. That was a very, very cool, eye-opening presentation. I really liked it. In the case of this kind of thing, I tend to go to a very biochemistry and biomolecular approach, because that's more of my, more of that I'm taught of. Like, for example, in the terms of aging, there are several factors that leads to the decline of immunological responses in aging. And a lot of those responses are influenced based on the methylation status. Now, methylation status and other epigenomic markers leads to, the downstream of that leads to the expression of certain proteins. Now, certain proteins, certain levels of expression can be analyzed through, in addition to the methylation itself, is through transcriptomics, which is now made possible through different methods on the same platform. Now, in the far future, well, not far future, but in the future, the idea is to find a way to be able to modify whichever status that is modifiable. For example, in terms of transcriptomics, we know, in terms of the transcripts, we know that there is a way to block those transcripts from becoming proteins, from becoming translated into proteins. And those kind of things are interventions that, in the vision of it, will enable interventions that either delay or reverse aging. That's the science fiction part of it, and the steps by steps are now available. Now it's about combining all those techniques and finding out how we can, I guess, connect them and find the right interventions for it.
Okay. So what do you think is the next step that every one of us can do here to figure out?
Right. This is a very loaded philosophical question, Dr. Mutia, that requires a group discussion and a focus group. But I think intuitively, I think we all understand here that scientists in Indonesia are the most efficient scientists in the world, because we only have a limited amount of resource and a limited amount of time to produce as much papers as international researchers. But to combine all that is how we can think about different steps leads to the same destination. For example, the current one of the current grants that I'm managing now is to analyze the effects of methylation for the immune responses in psoriasis, which is skin disease. Now, there is no way that I would be able to analyze the different kinds of psoriasis, for example, because there are different types, and there are different levels, and there are different severity. But my speciality is more on the analyzing why in certain cells the keratinocytes are, for example, affected by the innate immune responses that leads to them to infinitely replicating, creating the psoriatic lesions. So that's more of how I can connect the genes itself and then the transcripts that come out of it and how the proteins affect that. But how it's phenotypically presented requires a large amount of collaboration and trust and connecting the dots, basically. So that's what hopefully that we can do in this room, is that, you know, we can find a group of people that are of the same story, but they may be talking about different pages of this journey.
And and stitch it up together.
Yeah, and bundle it up together. Yeah, that's a good
Yeah,
one.
that's very inspiring. Thank you, Dr. Arya. And now maybe more onto a technical question. So we now, I know people in this room maybe have a mix of usage of minion or grid ion and promethion. So how do we actually determine which type of technology or flow cells do we need to use when we want to do human sequencing?
Oh, okay. Thank you for the question. I think the first time we need to know is the purpose of the research first. So, like, if we wanted to running whole genome sequencing or targeted sequencing or amplicon sequencing, the output or data generated will be different between them. And promethion flow cell is actually 5 times bigger in capacity than the minion flow cell. So we need to calculate the like a genome or target or amplicon that we need to know, and the capacity of the flow cell itself. So we need to ensure that we need to ensure that the data generated from the flow cell and the device, sequencer device, is sufficient for the next analysis by informatician and etc. Yeah.
Okay, so that's a bit of a calculation tips there, that the promethion flow cell is 5 times greater in capacity than a minion.
Yeah,
so
sorry.
we can choose our applications based on that. Any more questions from the audience? Yes, please. A lady at the back in black. Thank you.
Hello.
Would you like to introduce yourself first?
Hello, thank you for the session. My name is Farhani. I have like more general questions. My sister, 37 years old, she used to diagnose with myositis, autoimmune diseases a few years ago. After, like, we have no idea what the problem with her. She has like muscle issue, weakness on her muscles. And after getting diagnosed with myositis this year, she finally got a proper diagnostic, getting genomic testing. She has a rare disease, FSHD, which is like mutation in the UX gene or something like that. Currently, she wants to focus to test their children, want to make sure is it inherited or not. Is it possible to get a correct diagnostic for their children? Because we don't know in the future they have many, like there are many rare diseases like FSHD in the future. Is it possible to at least like predicting what disease can affect their children? Thank you.
Thank you. Just to clarify that question, is it like would it be predictive testing, or in the parents or in the children?
The parent already get proper test FSHD. Yeah,
So
that's
it
why.
would be done on the children.
For the children, because she used to get a not a correct diagnostic before. FSHD is the correct one. So is it possible to like predicting for the kids to make sure that they didn't get any like wrong treatment or something? Thank you.
Okay, thanks for the question. So the question is it possible to get predictive tests on the children or on the offsprings of patients with that? Ira, would you like to take that?
Yeah, thank you for the question. It's FSHD is actually one of the disorders that we are working on and using long-read sequencing to build better diagnostics for, because it is a very challenging genetic disease to diagnose because of the particular DNA sequences involved. They're very repetitive. So long-read sequencing is going to have a big impact in this area. And I think that the point you raised is really relevant to kind of all of these disorders, these rare inherited disorders that we're working on here. Many of them don't currently have therapeutics available to cure the disease, but that doesn't mean that a genetic diagnosis is not very useful for the family, both for sort of managing their condition and the onset of the disease, but for family planning and for organizing testing for potentially affected family members, because we know about we understand the inheritance patterns of these diseases. We can then predict, probabilities of likelihoods that children or other relatives may be affected, and then the family can make informed decisions about whether those family members want to have testing. It's important that it's important that this is still a choice for the family. People are not sort of forced into having genetic testing, but they can be informed about the risks, and that can help them make good decisions.
Okay, thank you. So it's definitely possible to do tests on the offspring or in the children, but most importantly, they have to maybe consent or agree or inform about that. Okay.
Yeah, and I guess it's important for clinicians and geneticists involved to counsel the families on their choices, given a diagnosis of a genetic disease, what the implications are, and make sure that they can make informed decisions.
All right, thank you. Any more pressing questions? Yes, Ibu Ines.
Hi, I've got a question for Ira. Given that your lab works works a lot on using genomics for clinical diagnosis, I was wondering if you could share a little bit of your experience on how your lab, which is at, I guess, the technical side, interacts with the clinicians first, and then maybe also give us a bit of an understanding on what sort of regulations apply to the use of genomics in a clinical setting in Australia? Thanks.
Yeah, thank you. It's a really interesting question. I should clarify that my lab is a research lab, not a diagnostic lab. So everything that we do is in a research-only capacity. We do not get sent, clinical samples and produce kind of diagnostic reports for clinicians. Everything that we do has to be done sort of collaboratively with a diagnostic lab, which are sort of state-based, nationally funded labs with very strict regulations on their processes and very tight quality control. So our job as a research lab is to kind of build the methodologies and capabilities that those labs can then potentially implement in the clinic. And so we're kind of like the R&D side, research and development side. We'll then sort of feed those processes into the clinical labs for them to hopefully implement. This process takes a lot of time. So the R&D side is hard, but the actual clinical implementation is very slow and rigorous, and lots of regulation that has to be navigated. And so there's always going to be a bit of a lag on the adoption of these technologies, but we have to sort of take a leap and start developing them if we're going to get there. And in terms of how we interact with the actual clinicians, the Garvan Institute in Sydney is a fantastic research institute that has a lot of partnerships with different hospitals, and we have a lot of clinician scientists embedded within the institute who study a particular cohort in kidney disease or ataxia or whatever, and these are the people who interact with patients and recruit them to our research studies, and it's really critical to have close collaborations with clinician scientists.
So because clinical implementation is slow and bureaucratic, in the meantime, though, like some of the results or some of the tests that you've done, you communicate with the clinicians, and that also underpins the patient care as well, right? But with the understanding that it's done in a research setting.
That's exactly right. So we can't, I guess, a clinical lab could issue a diagnostic report for one of these patients saying this is your genetic diagnosis definitively. We can't do that, but we can talk to the clinician who's managing that patient, and we have very close relationships with them. We can explain the data analysis that we've done, all the checks and controls, and how confident we are and what it potentially means for the patient, and that clinician can act on that information. They can, if they know us well and they know the technologies well and they trust the results, they can manage the patient's care accordingly, even though it's not an accredited diagnostic result. And so this is possible on a very small scale because it involves very close interaction with the clinician. If we want to sort of grow this to a national level thing that's going to see hundreds or thousands of patients, then it really needs to be going through this diagnostic lab where you just get a report and that's the result.
Thanks.
All right, thank you, everyone. And that marks the end of our first session. Thank you for our wonderful speakers, Ira, Dr. Ariel, and Himawan. I now hand over the event back to Raklia. Thank you, everyone.
All right, thank you, Dr. Mutia, Dr. Ira, Dr. Ariel, and Himawan for the stimulating first session. Before we move on to the next session, though, we are going to have a 15-minute coffee break. So please enjoy the snacks outside, and please return to the room around 10:40. So yeah, we will see you again soon. Please enjoy the snack, and good morning.
Selamat datang di BB Binomika, Balai Besar Biomedis dan Genomika Kesehatan, unit pelaksana teknis dalam layanan biomedis dan genomika kesehatan di Indonesia. BB Binomika bertugas untuk mendorong percepatan implementasi layanan berbasis kedokteran presisi melalui beragam program, di antaranya Biomedical and Genome Science Initiatives, layanan laboratorium kesehatan, penyelenggara nasional pemantapan mutu eksternal, serta sentral penelitian klinik di Indonesia. BB Binomika didukung oleh 6 instalasi unggulan. Instalasi laboratorium kesehatan klinik, instalasi laboratorium mikrobiologi, instalasi laboratorium kimia kesehatan, instalasi laboratorium biobank, instalasi laboratorium sequencing, dan instalasi laboratorium bioinformatika. Setiap instalasi kami didedikasikan untuk menjaga mutu dan menghasilkan pengujian yang akurat dan efisien, didukung oleh infrastruktur berstandar internasional. Bersama-sama, marilah kita membangun pelayanan kesehatan terpadu untuk Indonesia Emas 2045.
Nanopore Technologies, we provide top-tier genomic solutions, comprehensive services, and specialized training. Our commitment to national international accreditations and compliance ensures your scientific progress and discoveries with first-class solutions and direct hands-on experience for scientists, clinicians, and students. Visit genomica.id to obtain a quality genomics consumables and equipment online. Genomica Indonesia is your gateway to cutting-edge genomic sequencing solutions in Indonesia.
Take.
Selamat datang di BB Binomika, Balai Besar Biomedis dan Genomika Kesehatan, unit pelaksana teknis dalam layanan biomedis dan genomika kesehatan di Indonesia. BB Binomika bertugas untuk mendorong percepatan implementasi layanan berbasis kedokteran presisi melalui beragam program, di antaranya Biomedical and Genome Science Initiatives, layanan laboratorium kesehatan, penyelenggara nasional pemantapan mutu eksternal, serta sentral penelitian klinik di Indonesia. BB Binomika didukung oleh 6 instalasi unggulan. Instalasi laboratorium kesehatan klinik, instalasi laboratorium mikrobiologi, instalasi laboratorium kimia kesehatan, instalasi laboratorium biobank, instalasi laboratorium sequencing, dan instalasi laboratorium bioinformatika. Setiap instalasi kami didedikasikan untuk menjaga mutu dan menghasilkan pengujian yang akurat dan efisien, didukung oleh infrastruktur berstandar internasional. Bersama-sama, marilah kita membangun pelayanan kesehatan terpadu untuk Indonesia Emas 2045.
Genomika Indonesia is your getaway to cutting-edge genomic sequencing solutions in Indonesia, offering a wide range of products and services for research, education, and clinical testing. As a leading innovators in genomic industry and an authorized distributor of Oxford Nanopore technologies, we provide top-tier genomic solutions, comprehensive services, and specialized training. Our commitment to national international accreditations and compliance ensures your scientific progress and discoveries with first-class solutions and direct hands-on experience for scientists, clinicians, and students. Visit genomica.id to obtain a quality genomics consumables and equipment online. Genomica Indonesia is your gateway to cutting-edge genomic sequencing solutions in Indonesia.
Selamat datang di BB Binomika. Balai Besar Biomedis dan Genomika Kesehatan. Unit pelaksana teknis dalam layanan biomedis dan genomika kesehatan di Indonesia. BB Binomika bertugas untuk mendorong percepatan implementasi layanan berbasis kedokteran presisi melalui beragam program, di antaranya Biomedical and Genome Science Initiatives, layanan laboratorium kesehatan, penyelenggara nasional pemantapan mutu eksternal, serta sentral penelitian klinik di Indonesia. BB Binomika didukung oleh 6 instalasi unggulan. Instalasi laboratorium kesehatan klinik, instalasi laboratorium mikrobiologi, instalasi laboratorium kimia kesehatan, instalasi laboratorium biobank, instalasi laboratorium sequencing, dan instalasi laboratorium bioinformatika. Setiap instalasi kami didedikasikan untuk menjaga mutu dan menghasilkan pengujian yang akurat dan efisien, didukung oleh infrastruktur berstandar internasional. Bersama-sama, marilah kita membangun pelayanan kesehatan terpadu untuk Indonesia Emas 2045.
Genomika Indonesia is your getaway to cutting-edge genomic sequencing solutions in Indonesia, offering a wide range of products and services for research, education, and clinical testing. As a leading innovators in genomic industry and an authorized distributor of Oxford Nanopore technologies, we provide top-tier genomic solutions, comprehensive services, and specialized training. Our commitment to national international accreditations and compliance ensures your scientific progress and discoveries with first-class solutions and direct hands-on experience for scientists, clinicians, and students. Visit genomica.id to obtain a quality genomics consumables and equipment online. Genomika Indonesia is your getaway to cutting-edge genomic sequencing solutions in Indonesia.
All right, welcome back, ladies and gentlemen, to the ballroom. I hope the snack was delicious, and I hope you guys have the energy back to follow through with the rest of our sessions today. So we will be moving on to the next 3 sessions for the seminar. So please join me in welcoming back our moderator, Dr. Mutia Kumaheri.
Thank you, Raka. So I think everyone's excited to start our second session today. We have an exciting speakers who will join us shortly. I'll just like to remind a few housekeeping items before we start. So we will have 2 speakers for the second sessions. At the end of the talks, we will have our Q&A session. So I would like to invite all speakers back to the stage to take the Q&A session. So that brings us to ask questions during the Q&A session for our live audiences here. For our online viewers, please type your questions at any time during the talk at the comment sections in Zoom and in YouTube, and we'll read them to our speakers during the Q&A session. We also welcome questions in Bahasa Indonesia. We will help translating them to our speakers. Kami mengundang pertanyaan dalam bahasa Indonesia, nanti bisa dibantu tim kami untuk menerjemahkan pertanyaan kepada pembicara. So for our second session today, we are honored to welcome Professor Gunadi. Professor Gunadi is a pediatric surgeon and consultant at the Faculty of Medicine, Nursing and Public Health, Universitas Gadjah Mada, Dr. Sarjito Hospital. He trained as a pediatric surgeon, but his expertise extends far beyond the operating theater. He is the president of Indonesian Society of Human Genetics and the president for the Indonesian Young Academy of Science, demonstrating his commitment both in clinical care and in scientific advancement. As the coordinator of the Biomedical and Genome Science Initiative for Precision Medicine Hub at Dr. Sarjito Hospital, Prof. Gunadi is at the forefront of advancing genomics in healthcare. His expertise extends to pediatric surgery, rare disease research, and academic leadership. The title of his talk would be Adaptive Sampling for Variant Detection in Rare Disease. Let us all welcome Professor Gunadi to the stage to give his talk.
Okay, Assalamualaikum warahmatullahi wabarakatuh. Thank you, Dr. Mutia, for your kind introduction. So, first of all, I have to make disclaimer, I am not the expert of ONT. So when Dr. Mutia asked me to present regarding the adaptive sampling, so I expect my talk only 10 minutes, but surprisingly, she asked me to give talk 40 minutes. And then, due to my knowledge and skill on ONT is very limited, so I adjust my talk to add experiences and challenges. And then actually most of my slide will be composed of from Mavis in the back, from ONT, and from ONT Indonesia, Miss Fania, who helped me to make this slide. So I asked them last week, please help me to make this slide because Dr. Mutia asked me to present in the next week. So thank you, Mavis and Fania. And then in the left figure is the President Jokowi, I think 2 weeks ago, announced the new building in our Sarjito Hospital, a maternal and pediatric tower. Hopefully, the patient services for precision medicine, particularly for rare disease, will be services in that building, hopefully soon. Next slide. I'm lecturer from Universitas Gadjah Mada. Usually I make this kind of outline so my student will understand what learning objectives should be achieved. But again, I'm not thinking you are all my students, but it will help me to understand by myself. Next slide. Slide. Okay, sorry. I think I can use this. Okay, I think because the participants here is very diverse, very advanced, just like Bu Ines, Ira, maybe I'm not sure, maybe some of us is very beginner, so I will start with very diverse topic. So when do you remember the double helix discovered in 1953, and then the Human Genome Project starting 1990, and NGS introduced in around 2005, and 2014 the causal genome sequencing reducing to $1,000 US from more than $1 million US previously using Sanger sequencing. And 2023, multiple genomic project and initiative underway across the world, including in Indonesia, just mentioned by Bu Ines. Thank you, Bu Ines and team. Give applause to Bu Ines and team. Next slide. Sorry. Yeah, this is what I think we already, some of us is very familiar with genomic medicine, precision medicine. Genomic medicine is part of evidence-based medicine. The evidence, the type of evidence is genomic. So it's called genomic medicine. And then so far already identified more than 7,000 rare diseases over the world, including in Indonesia. Another achievement is prenatal care and IPD, cancer, pharmacogenomic, and population genomic. Slide. And when we look back in several decades, maybe when I was medical student, I study using light microscope, and then the next decade using karyotyping, maybe 2 decades ago, microarray, CMA for rare disease, and then next decade, the introducing of NGS. That's the history of how genetic disorder approach until now. And when the approach according to the genotype, several guidelines have been determined, such as if we suspect structural abnormalities, we may use CMA. But I think now the guideline, it's already changed. Exome sequencing should be coming first. But previously, CMA is coming first for rare disease. And then for mitochondrial disorder, if we suspect the phenotype mitochondrial disorder, then we may use exome sequencing plus, etc. And this is what Bu Ines and team work very hard. And then tomorrow will be the 2nd anniversary of BGSI, Indonesian Precision Medicine Initiative, and already 10 hospitals. And then our hospital, which is Rumah Sakit Sarjito, has been assigned by Minister of Health to perform genomic analysis for rare disease or genetic disorder. And then what is rare disease? Rare disease is the frequency if less or fewer than 1 in 2,000 population. And fortunately, more than 80% have genetic background. It means we may use genomic approach for diagnose the disease, and hopefully for precision medicine in the future. And then in Indonesia, actually estimated 13 million people are living with rare disease. So around us, around 13 million, among 280 million. And then most of them affecting children, and then most of them onset during childhood. This is the diagnostic odyssey. Usually takes 5 years at least for rare disease patient to get diagnosis before introducing of NGS. And then now in Europe, there are already over 230 orphan medicine to support for precision medicine for rare disease. And the goal in the next 5 years, 100 new therapies for rare disease, hopefully in Europe. And then this is the most important, 2/3 family carers spend more than 2 hours a day on disease-related task. And then feeling the pressure also occur 3 times higher than us, general population. I think we have to make our voice for rare disease, at least in this room. And this what strategies for rare disease prevention, not only after birth, but also before the baby birth, preconceptional, because of the fetal cell-free DNA, we can check using an IPT approach. And then after the baby born, now it's very famous newborn screening, rapid genomic testing for NICU babies, but defect prevention until the elderly. And this is, I think, almost all in this room, all people is understand that the first generation of sequencing is Sanger sequencing, and the second generation is next generation sequencing, and the third generation is long-read sequencing. And this is the topic what the committee asked us to present today. Actually, this is not the very beautiful finding, just Dr. Ira, but this is what we have faced, what we have experienced, challenge during the last 2 years using adaptive sampling. I think the last year. Before we talk about adaptive sampling, let's learn again refreshing about long read. So long read sequencing, according to the read length of sequence, we may define very easily short read and long read. And then if long read can directly generate sequence more than 10 KB from native DNA, does not require DNA fragmentation for sequencing, and then the step is usually first DNA fragment broken down into larger fragment from 1,000 to 10,000 base pair in length, and sequencing adapter attached to the end of the DNA fragment. After that, the live prep loaded into the long-read sequencing platform such as ONT. This is the very nice figure to compare between how the difference between short-read sequencing and long-read sequencing. If you want to read more detail, you may check the publication in Human Molecular Genetics 2018 later on. And this is the brief explanation, what is advantage and disadvantage. Also, we have to remember that one approach always have advantage, but in the other hand, they always have disadvantage. This is very, very simple explanation, the advantage and this drawback of long read and short read advantage. Utility of long-read sequencing so far, I think previous speaker already mentioned several times, including for high-resolution genome assembly. So that's why more complete genome information, because when using short-read, they might miss several kilobase or several genome. And then for targeted sequencing, I think Dr. Ariel already explained the role of long-read sequencing in immune response, transcriptomic RNA, now the single-cell sequencing also, epigenetics, methylation, and clinical application. For instance, I think, very nice presentation and finding from Dr. Ira regarding the tandem repeat pharyngeal eye syndrome and spinocerebellar atraxia. This is slide from ONT. That's how the nanopore sequencing works. So they use technology, call it a nanopore, and then after the DNA coming through the nanopore, and then they will hamper the current, and then they will see the finding of DNA, the finding of ATGC nucleotide. And this is the advantage of the ONT regarding the rich information, including methylation status, etc. And this is the difference between one device among others, depend on how many nanopores include in the device. And this is also the how many flow cell included in the device. Okay, let's move to targeted sequencing, one of the advantages of long-read sequencing. In some sequencing application, the focus of study single gene, selection of genomic region, small fraction of genome sample. In this case, targeted sequencing is more efficient. And then targeted sequencing using nanopore technology can be achieved in several approaches, including adaptive sampling. Again, I'm not the expert of adaptive sampling. I also still learn about adaptive sampling. Adaptive sampling is kind of offer fast and flexible method to enrich region of interest by depleting off target, and target selection take place during sequencing itself. And then once the run commence, sequencing will begin, and due to the real time of nature of nanopore, it is possible to identify whether or not the strand that is being sequenced is within ROI, region of interest. If the read does not map to ROI, MinKnow will reverse the polarity and then ejecting the strand from the pore so it is able to accept the new different template strand. And then off-target strand are continually rejected until a strand from ROE is detected and sequencing is allowed to proceed. This is the principle of, the main principle of adaptive sampling. Okay, I think this is the last sentences. Actually, for the adaptive sampling, the target is less than 10%. However, I will explain later on regarding what we have experienced using adaptive sampling. This is the adaptive sampling brief explanation. And this is consideration for experimental design when we plan to use adaptive sampling. First, library input, and then sequencing kit, fragment length, buffer size. This is also what we have learned about adaptive sampling, the importance of buffer size. The size of buffer also affecting much more on the finding of adaptive sampling. The target region I mentioned earlier, less than 10%. And this is what we have experienced as a rare disease hub in Sarjito Hospital. And we already sequenced 18 samples using P2 Solo. And then we use targeted adaptive and exome sequencing. And because we are still learning by doing, sorry, Dr. Aira. At that time, we used more than 500 panel gene, because we combine between muscular dystrophy, cardiomyopathy, arrhythmia, epilepsy, infantile epilepsy, etc. Finally, we got panel more than 500 gene. And this is the first trial for DMD is very good, because using the adaptive only DMD. And the result is very good. You can see. And also the second, but the port occupancy around 80% is very good. And PAH sample also very good because only PAH sample. And the target panel is very, very limited, only 23 genes. And after that, we start another trial and error. This is once we try the first batch, only the MD is very good. Finding pH also good. And then this is the compilation of what we have done the last year. For the first batch, I explained earlier that sufficient sequencing yield because the pore occupancy is good, and then single plex, and like this. Batch 2 also good, sufficient. Starting batch 3 is very bad. And then batch 4 also not good finding. Batch 5 also not good finding. Batch 6 also not good finding. And then we discuss with the ONT, Mafis and Fania from ONT Indonesia. If we see, this is also our mistake, yeah, our mistake or lesson learned, that we check the qubit during the QC, the first QC. Somehow we miss this very bad QC. The concentration is very low, even not reach 100 nanogram per microliter, and then just going to light prep and then going to NGS. This is the four sample with the worst result of adaptive sampling. And then you see the coverage even does not reach 20x, even less than 10x, because of this raw data. And we discussed with Mavis last week and Fania, this is what we have to do in the next. We have to optimize the sequencing workflow and light prep protocol, load more DNA to flow cell to increase pore occupancy and boost sequencing yield, and then need to have enough coverage to not decrease the analysis threshold less than 20x. Issue that when using exome together with adaptive sampling is not recommended because by adding buffer region is already more than 10% of genome. This is also another suggestion. We add another reagent from Twist Bioscience, but still there is drawback, is not cost effective because then the final price will be the same as whole genome sequencing. Remember, we use adaptive sampling to be more efficient than whole genome sequencing. But when we use exome sequencing and added Twist Bioscience, the price will be similar to whole genome sequencing. And then suggestion from Twist, existing target sequencing panels such as dark gene, pharmacogenomic, will be cost effective. Again, however, our hub is rare disease. So somehow the exome sequencing is more effective to detect pathogenic variant. And then optimize the use of Twist West panel on ONT. Let's say 1 to 2 kilobase. And then the Twist have had some customer run short gene panels. We just discussed with principal of Twist Bioscience last night. So before this talk, I already asked the Twist Bioscience, the ONT, to make this slide. So everybody here can learn from our mistake. So we can run together for the next. Nanopore platform is better. Do not repeat our mistake. And then another suggestion from Twist is design a smaller targeted custom long-read panel that is more relevant to disease of interest. I think the example from Dr. Ira lab is very, very outstanding finding. And then, alternatively, design a smaller targeted customer panel with adaptive sampling that of interest less than 10% target region. This is what suggestion from the ONT. We may revise the wet lab, and or we may revise the dry lab approach. For instance, we may use amplicon base. Just we discussed with Pak Usna and Mbak Mai from Rumah Sakit PON and RSCM how they use the amplicon base for clopidogrel and Fromody. And then for dry lab, this is what we have discussed. I think the best is less than 1% of genome, not more than 1%. This is, sorry for promotion, the next year will be the Asia Pacific of Conference of Human Genetics in our city, Yogyakarta. You're very welcome for all of you. Next year is biennial meeting in Yogyakarta. Thank you very much.
So it is indeed a rare experience to be able to learn from previous mistakes. So it's definitely an excellent opportunity for us, for all of us here, to hear of our first adaptive sampling experience here in Indonesia. So with that, I'd like to move on to our second presenter for this session. We have here Dr. Tida Tip Wongsurawat, or known as Dr. Tip. She is the head of Division of Medical Bioinformatics at the Faculty of Medicine, Siriraj Hospital, Mahidol University in Bangkok, Thailand. She also leads Siriraj Long Read Laboratory. She has an impressive research background with over 105 publications. So without further ado, I'd like to welcome Dr. Tip to the stage. Her talk would be Optimizing Long-Read Sequencing in the Medical Setting.
Thank you so much, and thank you for having me here. Good morning, everyone. Yeah, please just call me Tip. So now I am the head division of medical bioinformatics at Siriraj Hospital, and also adjunct faculty at the UMS USA, and also consultant for Thailand USCDC and consultant for ONT as well. Okay, so I moved back to Thailand in 2021 and start my job at Siriraj Hospital. Siriraj Hospital is the largest hospital that has 3,000 beds, and also support R&D as well. They have internal fund, but we have to publish Q1 paper, yeah, to serve them. Yeah. So I start my lab in 2021 and create this name, Siriraj Long Read Lab, or LOL, because I want my team as like laugh out loud, something like that, when they work with me. So at the beginning, we have only 2 persons wet lab, as me and Dr. Pilun is a bioinformatician. After that, we hire more people, yeah, and focus on bioinformaticians as well. So the technology that we use, of course, Nanopore technologies, because we use this technology in US before we moved to Thailand, and as the new investigator, no one buy the expensive machine for me. Yeah, so that's why I have to use Nanopore. And also bioinformatics. And this is why I moved back to Thailand. Actually, I have permanent resident in Singapore. And I think, should I back to Singapore or should I go back to hometown, something like that. And then I think, oh, it's not easy to push this thing in Singapore at that time, what I think. And one thing about economy of scale in Thailand is bigger. It means that if I do something research and move to the diagnostic, for example, it impacts more people. This is what I think. Then when I moved to Thailand, I think which project I should do. Then I think about Genomic Thailand project. Yeah, that combine 5 areas: infectious disease, cancer, rare disease, pharmacogenomics, and also non-communicable disease. And when I work in US, I have experience on infectious disease, yeah, sequence microbe, and also brain tumors before I moved back to Thailand. That's why I start with the brain tumor and infectious disease. But in 2024, I found that pharmacogenomics is like low-hanging fruit, and we can start this one first. Okay, I think everyone knows Nanopore, right? It's like a sneaker size, yeah, machine. And the good thing is the maintenance cost, yeah. So we don't need to pay next year maintenance cost. And this is the idea of the cross-section of the Nanopore when we start the minion or minion. And then the sensor can detect the electric current. And ATCG has different characteristics and size. Then it's, when it moves to the pore, it creates the pattern. And this pattern can be translated into the ATCG, for example. So when I start moving to Thailand, I have no space because I moved to Division of Medical Bioinformatics, no lab space. That's why I work at my condominium. Yeah, you can see the espresso machine there that can denature the protein. So it's very hard to work in Thailand at the beginning. And then after that, I asked my boss, please give me the space. And then he gave me his office because he has 2 offices. And then we start using this room as the lab and office. So have meeting together with doing lab work. However, more than 100 people visit my lab, including Handoko from Jakarta as well, and he got shocked as well. Yeah. So it's very small. After that, executive people asked me to help. Then I asked him, can I get this room? Yeah, a new lab. Yeah, very nice, Chao Phraya River view. Yeah, and I used my retirement money to pay for the aircon, many things. Yeah, because it's wait long time for government to pay, you know. It's document, something like that. That's why, okay, just pay. Yeah, even I don't want to pay, but I paid. Yeah. So now in my lab, we have many devices. Yeah, starting from the small one to the big one. Yeah, and I got support from Nanopore as well. Yeah. Okay, so this is the medical department. Before we make a decision to purchase the machine, we have to think many things, right? For example, Department of Microbiology asked me which device. Yeah, other department as well asked me. Yeah, so I asked them, what is the problem? Yeah, it's not about the budget. What is the problem? You want to sequence human or sequence bacteria or virus or something. Yeah, and then it's called sample type. And then after that, I asked about the budget and have to think about the budget next year for the maintenance cost as well. Yeah, not just 1 year and then next year have to return back the device, for example. And also scale. You got 3 samples a week or you got 100 samples a week. Yeah. And also portable. Yeah. So for portable, I have some video to show you. And after that, I list all the things for the budget below 3,100 USD. Yeah, but I'm not sure in Indonesia. Yeah, maybe the price may be different. Yeah, but in Thailand we have to pay tax. Oops, and margin for the company. That's fine. Yeah, let them clear the custom for us. Yeah, yeah. So this is what I learned. But if people ask me to set up Nanopore lab, I think 300,000 USD is better. So we have more device, better device. And also you have to think about the storage as well. Yeah, data is very huge. But if you have more, 1 million USD, you can purchase the robot to reduce the human error and some other machine, the size selection, something like that. Okay, and today I would like to show some case studies, what I use Nanopore in my department. The first one is full-length 16S RNA sequencing for pathogen detection. And the second one is metagenomics with adaptive sampling. Yeah. And third one, pharmacogenomics, cancer, and education. So this one, I present this work in London Calling last year about to use 16S RNA sequencing. The idea is the preterm birth, we need very quick result. Yeah. And one thing is about infection. So there are many reasons for the preterm birth. For the infection, the clinician, she will check about the interleukin 6. If increase of the interleukin 6, then move to the bacterial test. The good thing of Nanopore is real-time data analysis. After 5 to 10 minutes, we got the read, and then we can map the read to the database. So the current method for the amniotic infection is gram stain, and also try to culture the bacteria. Yeah, and sometimes some bacteria cannot be cultured. And also some people use Sanger. But you can imagine if we have polymicrobial infection, it's quite hard to read from Sanger. That's why in this case, Nanopore help. And we compare 3 methods. Yeah, the first one is culture. The second one is Sanger. And the third one, Nanopore sequencing. And you can see it takes only 5 to 9 hours to get the result. Yeah. But the price is higher than Sanger sequencing. So our finding, we found something like non-culturable bacteria called ureaplasma, for example. And also we can detect polymicrobial infections and directly from patient specimen. Don't need to culture the bacteria. Yeah. Rapid turnaround time at 6 hours. And this is the first study of intra-amniotic infection. So, and we publish in short communication format. And then I told this thing to ID clinician. And then they asked me back, when I detect ureaplasma and what's next? So I asked this question back to the clinician. And she said that, okay, she changed the treatment because previously she doesn't know what it is. Yeah, she knows after maybe 4 days, for example. And then not many labs in Thailand and USA growing this kind of bacteria. Also, at least at Sirat Hospital, clinicians need to ask or request to grow this kind of bacteria. So not only pathogen detection, some people want to study vaginal microbiome, to see the environment of the two pregnant, something like that. And also skin microbiome. And before I start using Nanopore, I have to purchase the ATCC microbiome community, mock community, and test on Nanopore. Yeah, I will use the primer to capture this bacteria first. Yeah, sometimes we miss some bacteria. Yeah, and we have to adjust the base on the primer as well. For example, gut microbiome. Yeah. So after that, we try not 16S because the question from clinician is, can we report the antimicrobial resistant gene? And 16S cannot say that, right? So that's why we try metagenomics with adaptive sampling. So for the short read, what happened? It's like small jigsaw, 100 to 300 base pair. And then it's, let's say this is the database that contains E. coli and Salmonella. The short read can map both organisms, because the jigsaw is very small. But for the bigger size, the long read, it can map only one bacteria, maybe Salmonella, for example. It's been that the taxonomic classification of the long read is better than short read in this case. And adaptive sampling that Dr. Ayla and everyone show here. So we can in silico deplete hosts from this case. We put human telomere to telomere data. And then if we sequence human, just push it out. But if not human, just sequence. Yeah. And we don't know what it is. Just any human, just remove. So in this case, we got sample. And it's abscess from this infant. And we found gram stain. This is very important that I make a decision to use this method. If I find only one cell, I will not use adaptive sampling because the amount is too low. Yeah. So this one, okay, maybe above 10%, above 1%. Yeah. Because when we get the sample, DNA of human also a lot. And DNA of bacteria as well. Yeah. So in this case, we use adaptive sampling. We extract the DNA. And the kit that I use to extract the DNA combine chemical and mechanical to break the cell, even positive, gram-positive or gram-negative, we can break the cell. And then let's say this is human mixed microbe DNA. And we do the library preparation using ligation kit. After that, we use gridiron because gridiron contains GPU, and we can remove human in real time. After 15 minutes, we cannot sit down. We want to know which kind of bacteria. And we went to the sequencing machine and download the data and map to the database. Yeah. And we found Prevotella. And sometimes if we do the same thing with short read, we can get many genus. But for long read, we got one genus, but many species. The question is, which species is true? We did the genome assembly of the bacteria after 2 hours. You can see here in the orange line, sequencing without adaptive sampling. Just sequence, get the sample and sequence. After 9 hours, you got 20x coverage of the genome. But if we use adaptive sampling, after 2 hours, we got whole genome sequence at 20x coverage. In this case, we found that we found the antimicrobial resistant gene as well. Yeah. So, and then we published in a good journal. Next, metagenomic of degraded DNA. For the degraded DNA, we cannot do adaptive sampling because it's too short. Yeah. In this case, what happened? The patient died after diving for fish, 5 days. Yeah. And then my collaborator, he do some 16S, and he found that, okay, this is erysiperotrix, which is maybe from pig. Yeah. And then, but he said that the wound, the thing is not the same. He want to know which species and AMR as well. But the DNA he gave me is, it's really short. It's like short read. So the size is about 400, 500, something like that. And you can see, no band, no high molecular weight band, right? And my postdoc, he told me the qubit is 3 nanogram per microliter. And also, in total, we have 72 microliter. And I said, we cannot say no to this person. He purchased us PCR machine. Okay, we have to do for him. So the good thing is, because Nanopore, we can do short fragment sequencing now. Yeah, just last year, maybe they released this feature. Even 20 bases we can sequence, or million bases we can also sequence. Then just do it, yeah. Use the ligation sequencing kit, not using the rapid sequencing kit because it's cut again the DNA, right? So we use ligation kit. And after that, we throw the data to EP2Me and also Chan Zuckerberg because it's free. Yeah. And then we did not expect anything, just do it. Yeah. And no exciting thing, yeah, just throw. And tomorrow we will see the result. If negative, just say negative. Yeah. We do our best. But we got surprised. This bacteria came from fish. Yeah, and the patient died because of diving for fish. And then we asked, is this real or artifact? Yeah, two databases show the same thing. And we found one publication at least support us. Okay, good. Yeah, this, this one, and this is high impact factor, is about 12. Yeah, to show that this bacteria can infect human. So, okay, good. Next, we move to brain tumor classification. Brain tumor is a type, brain tumor grow from glial cells, the cell that have function like a glue. So the mortality rate is very high and also current at, occur at any age. If we can classify patient, it's good for management, yeah, and treatment. You know, when I moved back to Thailand, when I work in US, WHO 2016, the guideline. But when I moved back to Thailand, they update the guideline to include the CDKN2AB deletion. Yeah, the marker, the new marker for classification. Then I got support from my boss. He said that, please help pathologists. Yeah, because now in Thailand, at that time in Thailand, no service for CDKN2AB deletion detection. And if we use fish, it's quite expensive, 500 USD. And also have to optimize to set up. So we set the goal to test the CDKN2AB deletion and reduce the turnaround time. And this is the diagnostic algorithm of the brain tumor, starting from, but this one starting from fresh frozen sample. And then they will check the isocitrate dehydrogenase mutant or not. Okay, and then they will check the copy number variation, and then they can classify the tumor into many groups. So in this case, I have to compare with fish epic array from Illumina and also whole genome sequencing from Illumina as well. Because this is a new method, yeah, we have to prove that our method is good enough. So I got 19 samples from my boss. He is a neuro-oncologist. Then before I start, I purchased the DNA that I know the chromosome 1 and chromosome 19 deletion. Yeah. And this DNA we test and use Nanopore to sequence. But for me, I want to do better than Nanopore. Yeah. So the original one, after 8 minutes, cannot see anything. But for our protocol, after 8 minutes of sequencing, we start to see the pattern of the deletion of the 1P and 19Q. Yeah. Then this is a real case. So we found that this person 1P and 19Q deletion. So in this case, we can classify this patient as the oligodendroglioma, WHO grade 2, yeah, 1P, 19Q co-deleted. And then we published a paper into the brain pathology. And we took 1 month to answer the question of the reviewer. Many questions, yeah, and looks like reviewer number 1, he loved fluorescent in situ hybridization. Yeah, so, and he said that pain point of Nanopore is bioinformatics. Yeah, something like that. That's why I said, no worry, we are developing a software. Yeah, that the clinician can throw the data, FastQ, yeah, into the Glimmers, our software, and then got the report, something like that. Yeah, like a web service. And we published this program, yeah, for free in Bioinformatics Advanced this year. So this is the idea that I want to make it happen in Thailand. After we published, my boss asked me, can we do the service? And then I asked him, can you sign for me? Because you are the clinician. If you can sign, I can open the service. He said, okay, maybe you can try 100 sample more. Yeah, and then he will make a decision to open the service or not. The idea is starting from 9 a.m. and then do some wet lab work, and load the flow cell, go for lunch. After that, my boss get the result, and actually we can wash the flow cell to use for the second person, second cohort, something like that. So also just this year we collaborate with DKFZ from Germany. They use EPIC Illumina to detect the methylation. And I think this is a good opportunity, not only Thailand, but Indonesia as well. For Asia, only 2 countries join this consortium. And we plan to use Nanopore and compare with the Illumina to see the methylation pattern. And this year also, this is not my paper, other people's paper, but it looks very interesting to use FFPE sample to do nanopore sequencing. I tried FFPE sample in US, that collect for many years and maybe 1 month, try many sample. And the result is not good. That's why I never touch FFPE again. But after this paper released, I try again in Thailand, So we found good result, using FFPE, but flow cell die quickly. It means that we have to think to trade off. If we can get the result faster, or maybe use prometheon, something like that. I try to develop this thing now, to use FFPE because it's more impact. Many hospitals cannot get the fresh frozen. Next. Pharmacogenomics. Have you heard about this thing? It's about study gene and drug of that person. For example, in Thailand, before people use allopurinol, the drug for gout, they have to test HLA-B first. If anyone has HLA-B 5801, they cannot use allopurinol. But in Thailand, not only allopurinol, but carbamazepine and abacavir also, they have to test this allele first. However, this is for Thai. If Caucasian and Japanese, if they don't have this kind of allele, then they maybe have other allele. So in this case, I think, why not we just sequence? No need to use real-time PCR or detect one by one. 5801 or 5801 or not, 5701 or not, something like that. Just sequence 1 time in your life and get profile, something like that. Then we developed, again, purchase DNA from USA. And this DNA already tests HLA by 3 methods. Next generation sequencing, Luminex, and something. Then I don't need to pay for HLA typing for this sample. And then we develop HLA-B testing based on Nanopore. Just sequence whole gene. After that, we compare with the Luminex method and got 100% consistent. However, Nanopore provide more higher resolution in this case. This is in Thai language. The person I show, he is the key opinion leader for pharmacogenomics in Thailand. And he found that PCR, SSP, the single specific primer compared to the hybridization provide different results. And he needs some method, sequencing method to prove that. That's why he sent sample to us, blind test, and we opened the result together. And we got a good result. And this is my friend. He has to use allopurinol. And then the doctor said, you have to test HLA-B5S01 first. And then he told the doctor that my friend can do sequencing. Yeah, don't need to pay the hospital. But you know, if come only one sample, it's more expensive. So, however, just proof of concept. Yeah, he sent me his DNA. And his genotype show like that. I sent him, you have 5601 and 2704. And he said, and then what? Yeah, so, oh, yeah, this is not a good report. That's why I put everything there, that, okay, you have low risk for allopurinol, carbamazepine, abacavir, and dapsone. Yeah, and I told him that he can keep this data, just use for any hospital. Yeah, and I call it HLA-B passport. And you know, if we sequence HLA-B, we can get all of these, can match to these drugs. Yeah, not just 4 drugs. Yeah, that's why sequencing is very powerful in this case. And I try to bring this to the hospital. And how about Indonesian? We found that 1502, a lot, about 11.6%. But I'm not sure you use carbamazepine or not. Yeah. So in this case, I think it's good to learn in each country, the frequency of HLA as well. And I present this work in Singapore, and I found that Tan Tock Seng and also Singapore General Hospital also interested to use this method. And just 2 days ago, before I fly here, so we just signed agreement with the lab service. And this lab service called nHealth. nHealth take care about 58 hospitals in Thailand. And they want to replace the PCR using Nanopore. And this is for the first time that we integrate Nanopore into the clinical service. Yeah. So, and also I asked for some budget to expand this thing to other countries. If you are interested, I think if I get the money from government, I will open for the training. So next we move to the education. I think the good thing of Nanopore is about portable, and education. So you can see we can train many people. And now it's above 1,000 people that we provide training already. And many countries as well. Also in Indonesia, just this year, we train 14 people from Indonesia. And this is my lab. We train in our lab. Also, this is the division of medical bioinformatics in Thailand. And also, we plan to go to Germany and Italy to training next month as well. In summary, I think for core facility, if you decide to use Nanopore, then you can answer many things, not just genomic, transcriptomic, and also epigenomic, microbiome, and metagenome as well. So I hope that I answer this kind of question. And if you have anything want to contact, just maybe leave your email or my Twitter. Do I have time to open the video? Could you please click the video for me? And this one, I use Nanopore to, in the field, to study the fecal, elephant fecal microbiome. So at that time, I work in US, and I follow many experts on Twitter. And I think that how can I have something Thailand-based? Not just UK or US, because I follow UK and US people. And I think that, this is Minion or Minion. And then I think about, in Thailand, the popular things is elephant. That's why I think maybe we study elephant microbiome, something like that. Yeah. We went to elephant village and we found the target. The baby. Yeah. Yeah, and we asked this person about how the elephant eat. Yeah. And I asked him, why not this one? He said, this have to buy. Yeah, and we try to find the lab part, lab space, yeah, and no electricity. So we use AA battery, yeah, for the spin down. And this is for the first time that I found fecal and feel happy. Because it's very fresh. Microbiome is very fresh and real microbiome. Yeah. So, and then we treat him this one. Yeah. And this is the extraction. Yeah. We use the beating method. And the machine. And when we do lab work, the baby want to join. Yeah. Yeah, and we have to use internet as well, use from mobile phone. Yeah. At the time that we do sequencing, we have time to do Thai massage as well. Yeah, and gave him for the supplement, yeah, to buy the sugar cane for him. Yeah, so this is what we use Nanopore in Thailand. Yeah, and I hope you enjoy my talk. Thank you so much.
Dr. Tip, please join me on stage along with Prof. Gunadi for our question and answer session. I would like to invite the audience for first 3 questions, and also online audiences can also ask their question on the comments section on Zoom or YouTube. So first 3 questions.
Dr. Mutia, we actually have the first 3 questions online already.
All right. Okay.
So maybe to switch it up a little bit, but the first 2 questions are from the same person, Mr. Firman Pratama Idris, directed for Prof. Gunadi, actually. So it's also on the screen, but it reads, excellent presentation and promising plan for rare disease diagnosis in Indonesia. While it looks promising, there are current barriers to using long-read sequencing for identifying variants in rare diseases, such as its prohibitive cost and the massive amounts of data it generates. Therefore, it must be targeted to individuals who need it most. How do you currently decide which samples are prioritized for analysis using long-read sequencing? This is the first question from Mr. Firman Pratama Idris.
Prioritization, so who is entitled more
Okay, I think this is also my question to Dr. Aira. Previously, right? Dr. Aira also already answered very well that depend on the type of target of the disease, the phenotype. If the genotype is very clear, several genes involved in such phenotype, then it's better use the adaptive sampling. However, if the phenotype and one phenotype have a very broad genotype and somehow we cannot pinpoint what gene involved in the disease, then we can use the long read sequencing. Again, that Sarjito Hospital still have challenges to improve after Dr. Aira presentation and Dr. Tip. I think we have more insight how to better design which method, whether we simplify, use the whole genome sequencing approach or adaptive sampling, or even combine exome sequencing with another such as Twist Bioscience to improve the quality of data generated.
Okay, thank you, Professor. Do we have next question online?
Yes, there's a next question from the same person. Okay. So the next question is, how does the Ministry of Health plan to provide the necessary infrastructure to support long-read sequencing in Indonesia, such as training or hiring of bioinformaticians, variant curators, cloud storage, and etc.?
I think Buina is more appropriate to answer rather than me.
Can we
mic-nya.
have a microphone for Ibu Ines, please?
Now, I don't know if there's, I mean,
Not to put you in the spotlight.
that's a very hot question.
Yeah.
So I guess through the establishment of this initiative, of the BGSI initiative, we have put the spotlight on sequencing, on genomics in Indonesia. And as a result, 10 of these hospitals now have sequencing capacity and biobanking capacity as well. We've established centralized data management systems as well. So that's part of the, how we've tried to set the seed. Obviously, we hope that this is like a ball that has to be, taken by the hospitals as well, and to improve on what has already been established. One of the things that the ministry is trying to do this year is to also finalize a genomic strategy for Indonesia, and that will also put a highlight on some of the things that need to be, the gaps that need to be filled, and part of these gaps include, the personnel, the skilled personnel. Bioinformaticians are very difficult to find, I think, in Australia as well, but, definitely in Indonesia. So maybe one way to do this, and, like Dr. Tip in her presentation, is to try and establish pipelines that can then be deployed by the hospital. So there's no need to actually do everything from scratch. And part of this is also in the system that's been established for the BGSI program, with its online data management system, and pipelines are deployed online, and obviously then all the different hospitals can point to that. So this is work in progress. Not everything is working yet, but we hope that over the next 6 to 12 months, obviously this capacity is going to be filled. So part of why we are doing this is to actually try and make sure that the benefits that are offered by genetics can be understood by the stakeholders and the people that determine where the money comes, right? And we need more people like Dr. Tip, who, she showed how, she started in her kitchen essentially, right? So we need to have stories to tell, so that we can also convince, the stakeholders to open the purses and give, more money to the programs. And I think we're going to see this, right? Some of the hospitals are already starting to have, stories that they can tell on how this can make a difference to patients. But I think trying to bridge that big gap in capacity is through collaboration, because if you have to work and learn everything from scratch, it's going to take a long time, and that's why we're facilitating and having people like Dr. Ira and Dr. Tib here to come to this. So and I hope that the people that are in the room will use the opportunity to kind of pick the brains, right, like of the people that are here and that have had a lot of experience. So cloud storage, that's centralized as well. So next door, there's actually an AWS workshop. So the ministry is actually providing storage capacity because we also want to make sure that this data is stored properly.
Thank you, Ibu. So the Ministry of Health is doing entire effort to build all of these bioinformaticians, training, variant curation, and also data storage and analysis and biobank as well. All right.
There's another one
from
for Prof. Gunadi again. So this is from Mr. Wisnu Ajikunchoro. He's asking, is adaptive sampling also being used to identify new pathogen or rare mutation currently in Indonesia? And what is the biggest challenge in doing so?
What is your biggest challenge so far, Professor?
Yeah, of course. Adaptive sampling can be used to identify pathogenic variant, yeah. But I think not for, correct me if I'm wrong, Dr. Aira, but not for novel gene, because for adaptive sampling we already have several genes until 500, let's say. Then we might find novel pathogenic variant, of course. among 500, let's say. But it's very challenging if of course, among 500, then some not yet reported, of course, did consider as novel gene. However, we already expect that among 500 is related to one phenotype. That's the message. So like, let's say muscular dystrophy have 200 gene, and then it might be one gene not reported yet. Then it might be considered as novel gene then. The most challenge, I think, actually we made mistake that did not control the QC at the first beginning. That's the lesson learned. We do hope that you are not repeat our mistake. So we have to make sure the quality control of QC DNA at the first. So just like Bu Ines said to us, go or no go. And at the time we just go, right? And then suddenly we look back retrospectively. Oh, okay, the QC is disaster. And then after that, we realized that after 6 batch running in using adaptive and exome sequencing, and then we discussed with ONT, and then not only the dry lab, the challenges, but also the wet lab is very tricky one.
Yeah, so every step of the way, there's a mechanism, a checking mechanism that needs to be checked to be able to go through to ensure everything is in place. Okay. Thank you for that. Live audiences, ladies and gentlemen, you invite for 3 questions, I think.
Actually,
yes, the first one. We'll have plenty of time for all of the questions.
Thank you. My name is Justinus from BRIN. My question for Dr. Tip. Before we also performance research about HLA profiling in leprosy patient. We targeted in HLA 31 because this HLA we know leads to hypersensitivity adaptation, or this research we performance in Papua province, Indonesia, especially in a remote area. The method we're using is AlloTX from CardiX running in Illumina platform. But in your presentation, you also development the methods to in nanopore sequencing. So my question is how you develop these methods. It's also targeted sequencing or another. Thank you.
Yeah. So for the HLA A, B, C, for my publications, we combine 3 HLA A, B, and C together. So actually, you can download the protocol as well. I did not develop the primer by myself. I use other people primer, the published one, and then try to combine, but use different method from others. I use rapid kit, that's cheaper. So when I moved to Thailand, I found that when I purchase enzyme, anything, I have to think about the price. That's why, okay, I moved to rapid sequencing. We don't need to purchase third-party enzyme, something like that. So in this case, just amplify the whole gene, maybe 4 kb or 3.5 kb, and then just do nanopore sequencing. Yeah, it's quite easy. Yeah, and we can detect HLA A3101 as well.
Thank you, Dr. Tip. Thank you, Vice Deans, for the questions. We'll move on. I think someone has got a microphone already. Yeah, let's go ahead.
Thank you. My question is for Dr. Tate. Actually, similar with the previous question just before me. Maybe I have another additional question. And that, how did you bring that into the clinical practice? You use it as a routine examinations, and how pay for the cost of the drugs is included in the National Health Insurance of Thailand?
Yeah. Oh, this is very good question. Oh, it's not easy. I could say that to move to the real clinical setting. Yeah, so at the beginning, I tried to move it to Siriraj Hospital because I think everything I develop, I would like to ask Siriraj Hospital first. But based on government organization, the decision make longer. Yeah, that's why I go to private hospital. Yeah, and the private hospital, they also think about per day sample, the volume, the scale. Yeah, this is another question. Is worth to do 2 samples or not? Yeah, that's why I go to nHealth, the lab provider that combine many hospital. Yeah. Yeah. So just one lab for 58 hospital. Yeah, but also not easy to convince. Yeah. So I went to key opinion leader. Yeah, who knows what is good, what is bad, something like that. Over 15 years experience, something like that. And I would like to say thank you to Indonesian people that invite me to Yogyakarta. And I had time to discuss with him for 2 days. So he also joined the meeting. Yeah, because in Thailand we have no time to talk. Yeah, even 1 hour. Yeah, he is very busy. So, and we discussed here, and we found the obstacle in Thailand as well. Yeah, and how can we move this kind of thing? So it's something that when he has problem, he has inconsistency between 2 methods, and he asked me to do. I said that please, I do it for free. Yeah, just send me the sample. I can show that Nanopore can answer the right question. Yeah, and then when we have the question, the answer together, and got 100%. This is one thing that we show. Then I went to nHealth, and then I asked nHealth that please give me the hard one, yeah, the hard sample that cannot answer. And they brought me the sample that keep for 5 years. Yeah, the DNA is open the door of the refrigerator for 5 years. Yeah, something like that. Yeah, so to challenge. Yeah, and also some sample that the inconsistency between 2-3 methods, something like that. And we did that and open answer together and got 100%. Yeah, after that we try 400 sample before they make a decision. Yeah, 400 sample and get good result. Yeah, and then they think that, okay, even it's expensive than using PCR, but it creates value for people. That's why they make a decision to change, and they want to be like a private hospital who is like international hospital, not just for Thai, but for anyone. Yeah, foreigner as well. Yeah, so this is how I move. And then I think government will see this thing and open for this thing as well. I went to Thai NIH as well to support them. Yeah, when they have anything that inconsistency, I said maybe try Nanopore. Yeah, something like that. This is the way that I move. Yeah, so yeah.
Maybe on that final note, Dr. Tip, so who pays for that pharmacogenomic test?
Okay. Government paid. This is the reimbursement. Yeah, that's why I moved to the gene that have reimbursement first. But the reimbursement only 33 USD. Not all. Yeah. That's why we have to think about the volume per day. Yeah.
Okay, so does the reimbursement covers for the whole cost of the test, or is there any discrepancy that patient may have to pay?
So actually for government hospital, it's less than 33 USD.
So
yeah, but
margin.
Yeah, but for private hospital depends. Some private hospital create package. Yeah, because they think that this is better than just PCR test. Yeah, then they can create package and give to the person who want to pay, something like that.
Okay, thank you, Dr. Tit.
Yeah.
Next question. Yes, please.
Yeah, thank you for the time. I want to ask Dr. Tit.
Yep.
So right now we are doing metagenomic from MPMA specimen. As we know that some pathogen or some bacteria is normal flora in human, however, they can be opportunistic pathogen also.
Right.
So how we decide this bacteria is colonization, contamination, or this is a real pathogen based on our sequencing result? Thank you.
Yeah, this is very good question. And before I start this thing, I discussed with my friend in Singapore as well, in Genome Institute of Singapore. And there are a list of the bacteria, not just one bacteria, maybe 20. Yeah, but the thing is, the clinician will know about that. Yeah. So when we report, they will know, oh, Staphylococcus aureus or Streptococcus epidermidis, yeah, from skin, something like that. Yeah. So this work have to work with clinician. Yeah. This is the answer. And in Singapore, as I know, my friend said that has to do disclaimer. And they said that, oh, it's about A4 disclaimer. It's very long. Yeah, yeah. So I hope that in Thailand, we just start this kind of thing. And we work with clinicians. The good thing is I work in hospital. That's why we sequence and then we discuss. We got 30 samples from hemoculture as well. And we sequence the sample from hemoculture. And then the clinician said, okay, this is real. This is just from the skin, something like that. Yeah, we also learn with them. Yeah. Yep.
So somehow we found bacteria with less number than another bacteria, the abundance. Is it possible that this bacteria also can be pathogen, or we can ignore this case? Thank you.
It's possible, yeah. Sometimes the high abundance may be because of the environment, the pH change, something like that. Maybe not pathogen as well. Yeah, yeah.
Other questions?
Yeah, actually I have 4 questions, but looks like other audience want to ask, so I just bring up 2 questions first. My name is Lawrence. I'm from Faculty of Medicine, Chulalongkorn University. So, Sawadee-krap, Achan.
Sawadee-krap. Welcome.
Yeah, so I would like to Achan Tidatip first. Back then, during I have already asked this question and have a small discussion with Achan Pirun about this. So based on my experience working with Oxford Nanopore sequencing, how do we identify if there is a SNP that heterogeneity or heterozygous? I'm working with G6PD deficiency patients from around, so I sequenced around 2,000 samples from King Chulalongkorn Memorial Hospital. So the problem is when we deal up with a G6PD deficiency, it's X-linked recessive, right? So somehow it should be like the heterozygous. But the problem with nanopore sequencing is the outcome of the consensus is the most common one, right? So, but yeah, I have a problem to identify the SNP. Actually, not in the term I cannot find the SNP, but in the regular way, when we use Assanger sequencing, when we send the sample for Assangers, they will show the chromatography, right? And show 2 alleles.
Yep.
But in nanopore, no. So back then, I do maybe like the conventional way. I talk to my boss, Professor Sanchai Payungporn. I believe you know him.
Yeah, yeah.
Yeah. And then he said, just go back to the BAM file and then see like if it is like heterogeneity in this in this row, in this column or not.
Yep. Yeah.
But I think that that's kind of subjective because Yeah, you know, right? Because it's
yeah,
based
yeah.
on our briefly see the result. So is there any
Criteria? Something?
yeah, to say that, okay, so this one is the heterogeneity. That's my first question. The second question is also work with thalassemia cases. Yeah, because thalassemia is high prevalent in Thailand.
Yeah.
The similar problems, thalassemia is deletion mutation, is not SNP, right? How do we identify if the patient have double mutations? Example like thalassemia 3.7 mutation and 4.7 mutation as well. Because if we sequence it one at a time, it will just show one outcome, right? The deletion. So how do we know that the patient have a double mutation? So we sequence twice, or how to address these issues? I think that's my question first. If have time, then I will ask the other question for Professor Gunadi. Thank you so much.
Yeah. Okay. Yeah. Very tough question. About the SNP, we found the same problem. If we use the PCR, sometimes the primer prefer to buy another allele. Yeah, and the SNP, for example, we should see 50-50 or 40-something 60, right? But sometimes we found 15, 85, yeah. And the software cannot detect that, yeah, because the software say that it's above 30. So we have to relax the software, yeah, to detect that. For example, for HLA, we try many samples, yeah, and then we check what happened. We know that this kind of sample heterozygous or homozygous, and then we open the BAM file, and we try to find the criteria, not 40, 60, never. I'm not sure you use CLAIR 3 or not, yeah, to identify. Then we can relax the criteria. In this case. But for thalassemia, maybe Dr. Pilun can answer better. We collaborate with Dr. Suthat, to amplify the whole gene, beta, alpha, something like that. This is just the first batch. We got good result for beta, but for alpha, it's not easy. So we try to move to the next step to optimize the tag polymerase, something like that. The condition, and to sequence it. Our experience on thalassemia just very low. Just start for us.
Okay, would Dr. Pirun like to elaborate on the thalassemia?
Okay, so according to your question, if you want to identify the double mutation, right, and if the region is very far apart, so it would be better that you can do whole genome sequencing or adaptive sampling of that region instead of the amplicon sequencing, because amplicon is not long enough to cover the whole thing. So that would be the way, I think.
Right, thank you. Does that answer your question? Yes, okay, thank you. We would have time for one last final question. Yes.
Thank you for the wonderful and insightful presentation, Dr. Tip. I would like to know more about metagenomic and adaptive sampling plus adaptive samplings. So in what situation or condition the metagenomic plus adaptive sampling approach will appropriate to be implemented? And also, what is the most important aspects that we need to pay attention when implementing it? Thank you.
Okay, thank you for the question. The one thing for adaptive sampling, the abundance, the amount of the bacteria or organism that we want to study. That's why I make a decision after I see the gram stain. And we have 2 more cases from heart valve. Yeah, they do gam stain, but the number of the cell is lower than this, maybe 5 times, but I still working on that. And I found that for the one that I show you, it take only 2 hours. Another sample is take 9 hours with adaptive sampling, depend on the DNA of the pathogen as well. This is one thing. The second thing is about the size of the DNA. Yeah, if we get sample long time ago that keep in the fridge and it's sheer, don't use adaptive sampling. Yeah. So this is should be high molecular weight. Yeah, even if this sample keep for a long time but in -80 and not freeze thaw, it's possible to use that as well. Yeah. But for blood, directly from blood, I never try and I will not try. I think it's not easy. It's very dilute. Yeah. So then, but if anyone try, please share it to me. Yeah. Not easy, I think. Yeah. I found one publication to say that the number of the bacteria that can be detected from joint, from local fluid is higher than blood. Yeah. That's why we have to think about that. Yeah.
Yeah, so sample source is very important in this case.
Right.
Okay. All right, so we're at the end of the second session today. Ira, do you have anything to add or to comment on? I think I saw you raising hand, or am I wrong? Yeah, microphone is coming. No, okay, we're good, okay. Okay, so ladies and gentlemen, that brings us to the end of the second session. We hope that you've had all of your questions answered, but otherwise feel free to ask directly to every one of our speakers, either like during the break, lunch, or in the afternoon tea later on. All right, so let's give a round of applause for our magnificent speakers, Prof. Gunadi and Dr. Tip. So I will directly move on to our third and final session for this morning. We'll be hearing from Dr. Seeley from Oxford Nanopore, especially Associate Director, Clinical Sales Specialist. He's based in Singapore, and he's taking care of in vitro diagnostic partnership and diagnostic testing status in Asia Pacific region. Dr. Seeley has years of experience in IVD assays and workflow development, system integration, and clinical market exposure. His work span across multiple biotech and genomic tech companies. We will hear from him of long-read sequencing solution for detection of drug-resistant tuberculosis. So please welcome Seeley to the stage.
I'm honored to be the lunch gatekeeper of this event. Yeah, so I know everyone is hungry, and I think the market is also hungry for ONT-based clinical applications. So here is one of it. So the name of our product is actually the MPO-TB. It's the first ONT's home-brewn IVD assay. So this is actually a TGS assay for the detection of MTB diagnosis. So it aims for provide the rapid testing of AMR resistance with ONT Nanopore sequencing technology. Yeah. Okay, maybe the pointer doesn't work, so I have to say next. Yeah. So this is an overview of the MPO-TB workflow. So MPO-TB is not just an assay, it's not just a software, not just a kit. So it's generally the whole integrated workflow. So several key metrics of this assay workflow is it just takes 6 hours all the way from sample to report. The sequencing time just takes 2 hours, and it generally profiles all the 16 anti-TB drugs screen recommended by the WHO guideline. And if you talk about the solution, it's one fully integrated solution. Yeah. Next. This is the journey how this MPO-TB has participated in the Seek and Treat program, which has been promoted by the WHO together with FIND. Next. So starting about 4 years ago, I think WHO have launched this program named Seek and Treat. So it aimed to employ the next generation sequencing or even other high throughput sequencing technology to tackle the TB problem globally. So it's also part of the NTB program globally. Yeah. So starting from then, WHO start to screen all those kind of high throughput sequencing technologies, and own nanopore sequencing definitely is scored and also be selected by after the evaluation. So as you can see from the screen, actually this the whole scoring and qualification process is a very lengthy one, and it's very strict and need a lot of qualification steps, a lot of data collection, a lot of testing trials. So now we're in the stage of stage 2 clinical evaluation. We have actually tested over 800 samples across 3 continents. So after that, the outcome is actually in this March, WHO actually released this consolidated guideline. So in this guideline, they recommend 3 products, 3 TB assay products. So the first one, Diplex GenoScreen, is from GenoScreen company, a French company, GenoScreen, employing the Illumina platform. AmpoTB is also selected as one of the 3 recommended assays, and this is developed by ONT ourselves. And the other one, the TB-Seq, is also developed based on the Nanopore sequencing platform. So it's a company in China. They're also our ONT's potential IVD partners. So generally, 2 products, 2 recommended products out of the 3 are based on ONT platform. So this is just the sample metrics, how these 832 samples are consist of. And some of the samples, they do have the WGS result, some do have the PTSD result. We actually compare with those samples which has been both the molecular method result as well as the traditional method. Yeah. So generally 685 samples has been consistent with the other technologies result. Yeah. So based on this evaluation result, actually WHO released both the consolidated guideline as well as the operational handbook for the customer to refer to. So those documents, official documents, can be found from WHO website. So if any customer interested, you can refer to this document for details. So this is just an overview of the whole AmpoTB workflow. So the breakdown can be a few steps, consists of decontamination, cell analysis, which just take about less than half an hour. And then the extraction purification take another 1 hour, and the multiplex PCR take 1 hour plus. Then with our just half an hour rapid library prep as well as a washing, we can apply the library onto our nanopore sequencer, which take about 2 hours for finish the sequencing. And again, the analysis and reporting take just maybe 1.5 hours. Yeah. So in total, it takes about 6 hours all the way from sample to report. These are the 16 anti-TB drugs which has been recommended in WHO official guideline. So our AmpoTB assay actually cover all the 16 drugs. Plus, we also cover the HSP65 target, which is aiming for the NTM identification, and also the CRISPR locus, which is more for the MTB identification, species identification, and another internal control. Yeah. This is the schedule which we design our whole workflow. So generally, one flow cell we recommend to run up to 24 samples. So for example, the first one you can employ the barcode 1 to 24, and the second one, in order to minimize the crossover contamination, so we recommend to run another totally different set of barcode, which is in this case like 25 to 48. And again, for the third run, you run for barcode 49 to 72, and for run 4, you run the rest of the barcodes. So in this way, you won't use any same barcode in the adjacent runs. So in this way, the cross-contamination can be minimized, and you can freely leverage the beautiful feature of our flow cell, which you can reuse our flow cells to reduce your operational cost by 4 times. Yeah. Okay, so I would like to dive a little bit into each of the steps. So for sample preparation is actually we are compatible with both the manual or automated extraction. So in this case, we actually give 2 examples. For automated extraction, our R&D actually we test against the Prometheus Maxwell platform. And regarding the manual extraction, actually there are many choices in the market, but internally we actually tested the Cargene DNA Easy Blood and Tissue Kit, which works really well. Yeah. And this is the breakdown of the sample preparation workflow. So you can, I think the most time-consuming step is actually the PCR setup and application itself, which cannot be avoided for the targeted assay. However, for the other ONT-specific steps, you can see the rapid barcoding only take 10 minutes, the pooling 5 minutes, and the purification just 30 minutes plus the adaptive attachment, which take 5 minutes, and then you can load your library onto the sequencer. After you apply for the sequencing, actually we totally redesigned our Minone software, which is our operating software. So this one we call it actually this DSS software, which is actually the toolset of the diagnostic version of the Minone. So generally, it's a more closed version. It's per the DX diagnostic standard, and it comes also with the user access control as well as the audit trail. So the user interface will be very brief and neat. So customer just need to load your library onto your flow cell, then you can choose which flow cell slot you choose. And then you need to probably choose the test. In this case, because we only have the AMPO-TB coupled in this, integrated in this software, so you can just choose TB test, and you click continue, and then they will do the QC check of the flow cells. After that, you can import your sample list, or you can manually input your sample information. Then you click next, then you can start the run and perform the assay. Yeah. So it's very easy, friendly use. And then after analysis, you after like 2 hours of sequencing, you can start to get the report, and then you can see the report actually consists of all the necessary information needed to make the medical decision. And you will have all the control metrics as well as the key metrics of the assays. So QC has passed, and so this is example of one clinical sample. So you can see on the left, you can see all the species, the spoliogeal type identified, as well as which drug is resistant based on the sequencing data. Yeah. And furthermore, in the detailed report, you could find all those SMP calling and any indel calling, as well as the resistant annotation recommended in the WHO guideline, including all the WHO mutation catalog numbers, as well as the expert tool rules and the full gene dropout information included. Yeah. So if you want to dive further into each of the samples data, so you also show the coverage data, as well as the pass or fail, and also you will also come with the whole integrated report together with every information integrated into one report. Yeah. Yeah. Okay. So for this AMPO-TB's launch, actually we have commercially partnered with bioMérieux. So I believe everyone is familiar with bioMérieux as an IVD company. So they have been founded 60 years ago, and this is was leading maybe IVD companies. So they also have a lot of channels, partners established across all the countries. So bioMérieux will be our official global distributor for the AMPO-TB assay. Yeah. So they will go through 2 routes. So generally for the public sector, the bioMérieux will generally they will support directly and will sell through some official platforms, purchasing platforms. Yeah. Well, for the private sector and commercial customers, so they will go through their channels. It's about to be established in the coming months. And for the AMPO-TB's official launch, together with bioMérieux's maybe commercial launch, is estimated in October. So please stay tuned, and all those kind of service support, pricing information will be available by then. Yeah, and the local logistics as well. Yeah. Okay. Okay. That's my brief introduction of the AMPO-TB. So yeah, now I'm open to questions. Yeah.
All right. Thank you, C. Do we have questions? I'll join you on the stage. Questions online or from the audiences? Just before lunchtime. Okay, I'll open with a question. How would the report integrate to maybe like hospital e-medical record?
Yeah, that's a good question. I think most of the hospitals they have to have their LIMS or maybe HIMS, right? So we do have the API, the API which is to can be integrated into based on the like a VL7, ASTR, the standard to be integrated into a LIMS system. So it's taken into consideration already. Yeah.
Okay. Ibu Ines, please, and maybe one microphone to Dr. Ariel, so we can move along. Yes.
Yeah.
Thank you. So Indonesia is at the moment in the process of utilizing ONTs, I guess, in-house
Yeah.
pharmaceutics
Yeah.
in about 3,000 samples all around Indonesia. So how different, and in the process, I think about 5 or 6 labs are being trained on that. So how different would it be to transfer from this in-house process to this diagnostic kit?
Yeah. We are aware that actually we do work with some Indonesian labs on our custom kit, we so-called custom kit, which is used to be the prototype of this ample TB assay. So technically the assay design is still the same. It's just with ample TB, we launch as more like a commercial product. However, for our custom kit, the assay design and the like the control design, the protocol is more or less the same. So we just finalize it and wrap it up as ample TB. And also, ample TB also consists of this reporting software, which we haven't provided us at the prototype stage. So you will be able to access this easy-to-use software upon launch. Yeah.
Okay, so the transition will be seamless.
The transition. Yeah.
Can I ask one last question?
Yeah.
With the software, so the software is on the machine, or is it on the cloud somewhere? And if it's on the cloud, where is it hosted? And the reason I'm asking is because there are a lot of, there's regulations in Indonesia about sending sequences, like information on overseas cloud. And if the cloud is not hosted in an Indonesian server, then there will be things that need to be set up.
Yeah, that would be a common concern for clinical setting, right? Yeah. So generally, right, this ample TB, by default, it need to be run by the cloud. But if per customer request, we can also make it locally with the local database. But this maybe need to install not only on our GridION, but maybe need to install on the workstation, maybe collect it to the GridION. Yeah. Yeah.
Okay. So because I guess the hope is to have this part of the national TB program and be used like around Indonesia. So aside from the technical aspects, we need to ensure that we are abiding by the regulations that have just been, established and launched.
Yeah, exactly. So at the same time, we have already submit the ample TB to FDA approval. So we also take this local, data localization requirement into consideration. So that would be a localized version. Yeah.
Localized version.
Localized version. Yeah.
Of the cloud or the analysis software.
Yep.
Okay.
Yeah.
Dr. Ariel, please.
Thank you very much, Mr. C. I brought the results of M4 TB testing kit with West Java Laboratory to London Calling. So thank you very much for presenting it again. And I guess my question is that during that London Calling, we presented, and there was another presenter from Mayo Clinic that was also reporting about M4 TB. And we found very similar challenges in terms of detecting some parts of the mutation. We found very similar success rate and failure rate on certain resistances. My question is that I don't know if this is a technical question, but how do you presume that this will be managed in terms of the distribution of that, for example, a localization kit for the analysis and also the kit itself? Are they going to be updated? And does that update requires a different kind of validation with WHO? I don't know about that, so could you please enlighten us about it? Thank you.
Yeah, that's a good question. I would say if there's certain, for example, higher dropout rate for certain target, that may be, there are several reasons. It may be due to the assay design, for example, the primer design. For this case, we may have to maybe upgrade, for example, in the next version of this assay. However, if that dropout or maybe that pattern may be due to something like more like a software, we probably can solve by the software upgrade. Yeah. So we need to further look into that issue if if you have feedback, better feedback, yeah. But I think we need to collect the feedback from different customers, and we want to investigate further. So it's either by the assay upgrade or maybe the software upgrade. Software may be easier, yeah. Yeah, hopefully, yeah, hopefully.
So what's the best pathway to send this feedback other than I mean, we can publish it, but what's the best way?
You can treat me as the regional maybe contact.
All right,
first
okay.
contact, yeah.
All right. Fantastic, thank you.
Yeah, you have my card.
Yeah, I do. Thank you.
That's perfect. Okay, any more questions from the audiences?
Dr. Mutia, there's an online question.
Okay, good.
Yeah, so
Go to the
this
online.
is from Ms. Triuli. She's asking about, is there any specific criteria for these sputum samples that's to be used for Ampere?
Maybe just normal sputum.
So
Yeah.
RSPESS is actually the hub, the BGSI hub, specialized for respiratory disease.
Yeah.
And I think they're utilizing the early access kits.
Yeah.
And I think this is real life questions.
Oh, real life.
Real life
Okay.
experience.
So I just, I mean, I will add to what Buyuli is saying.
Yeah.
So I guess they're just starting, and I've started running a few hundred samples, and I guess from these few hundred samples, some of them, and I can't remember the percentage, they can't get a result out of it, right?
And
so that's why I think the question is, what's the criteria of the sputum? Is the problem with the sample? Because if the problem is the sample, we need to actually give that information to the labs that are providing the samples.
Yeah, that may need some troubleshooting effort to make sure whether this problem really, the failure really come from the sample metrics or come from the. But generally, what I can share is actually not only from AMPO-TB side, we do have the seq and treat. We test maybe 800 plus samples globally, and we also have even from another product which I introduced just now, like the TB-seq from another third-party company named Shenting. Yeah, it's totally different design, but they tested over 8,000 samples globally as well. So I also have close contact with them. I don't think they think most of the failure came actually from the sputum sample itself. Yeah, but maybe I think we still need to analyze, maybe combine the data together to further analyze whether there is any specific requirement for the sputum samples together, because different customers, they may use different extraction strategy. Maybe they may use Cargin key, they may use some automated liquid robot. They also may need use some other like maybe other kit, extraction kit, like Zymo, like other kit. So yeah,
is there
it's
any metrics, any indicators of what a good extraction or a successful extraction is?
Yeah, I think generally we have our maybe a DNA quality, this kind of QC or QC method. As long as we meet our protocol requirement, that should be okay. Yeah.
Okay.
Yeah, I don't see there's any sputum specific requirement. Yeah.
Okay.
Yeah.
Right. Yes. Come
on. Okay. You mentioned earlier that this assay using rapid barcoding kits and grid ion device with min ion flow cell, right? Is it possible if users use their min ion flow cell on min ion MK1C or min ion MK1B instead of grid ion?
Yeah, it's possible. We do have some customer is doing that. And it just maybe if you use Milan Mark 1B, maybe your workstation need to be up to standard. And if you use Mark 1C, you may expect a slower, maybe base calling rate a little bit. Yeah, yeah, we do have customer do that. Yeah, but our official launch, maybe the platform is still the Gridiron. Yeah, yeah.
Thank you.
It's possible. Yeah.
All right. Up next question from the audiences or online.
Maybe the last question, if there's
one,
any.
Final.
Yeah, I can see everyone's hungry. Yeah.
All right, that we won't keep everyone from their lunches. Thank you, Silly, for
thanks for invitation.
introducing the solution to us. And so that ends our seminar this morning. I'll give the event back to Raka, who will lead us to lunch.
Yes, thank you. A round of applause maybe once more for Celia and Dr. Mutia. Thank you. All right, ladies and gentlemen, we have now come to the end of the seminar segment of today's event. Before we all disperse to lunch, I'd like to invite everybody to come to the front to take a picture together with the speakers, the moderator, and all of the facilitators. So the committee, can I ask for your help to organize the speakers and the participants? Let's go. So maybe the speakers, the moderators can be up on the stage, while the rest of the participants, maybe we can manage to put some on the stage and some in front of the stage. Yes. Okay, so maybe the participants can come up the stage, a few of you, so that we can see the proper ratio to take a picture. Yes, please, for participants can get up on the stage. And then the speakers, facilitators can be centered in the front. How does this look, committee? How does this look at the in the camera? Does it look okay? Do we need some of the participants down on the stage, in front of the stage maybe? How? Oh, okay, maybe we can make 2 layers. So for the participants on the stage, for the ones that are directly behind the speakers, can crouch a little bit so that everybody behind you can be can be seen. Maybe the tall or maybe in the front. How does it look? Okay. Just until the edge of the stairs? Okay. So maybe can... Oh, okay. So if some of the people don't mind, you can crouch in front of the speakers because we don't have much space. So maybe the ones that are on the edges of the stairs can come to the middle and then crouch a little bit if you don't mind. Or maybe the men, if the men wants to do that. Oh, Dr. Ayer, no need, please. Okay, does this look okay? Can we test it on the camera? Does it look okay? Okay, 1, 2, 3. Once more, 1, 2, 3. And on the count of 3 now, say long read. 1, 2, 3. Thank you so much, everybody. Oh, not yet. Oh, one more. Sorry. Okay, clear off. What is this, video or? Video and photo. Say sequencing? Wait, in genomics, we trust. No. How about we just wave? You know, like. Okay. Yeah, just do a little wave, everybody. Okay, this is a video, yeah. Is it already taking? Okay, keep waving, everybody. Thank you. Perfect. Okay, thank you so much, everybody. You may return to your seats as I will divulge the final announcements.
Good afternoon, ladies and gentlemen. My name is Levanya, and I'm gonna be your MC for today. We would like to welcome everyone to today's workshops on adaptive sampling know-how. We have an exciting agenda with 2 expert facilitators who will dive deep into the latest innovations and tools that make adaptive sampling a cutting-edge approach in genomic analysis. We also like to remind you that our team are ready to assist you throughout this workshop. So without further ado, I'm gonna introduce you to our first facilitator, which is Rebecca Chesterfield, that will facilitate us towards the introduction to MINOW and adaptive sampling, and also bed life design, testing, and operations of adaptive sampling. Rebecca Chesterfield is a technical application scientist at Oxford Nanopore Technologies that holds a PhD in life science from the University of Queensland. With over 10 years of academic research experience, Rebecca has become a vital resource for genomic labs across Australia, New Zealand, and also Southeast Asia. Her expertise lies in providing comprehensive technical support and driving forward application in cutting-edge sequencing technologies. So without further ado, let's give a round of applause for Rebecca that's joining us online today. Hello, Rebecca, how are you? You may start your presentation now.
Thank you very much. I'll share my screen. Thank you very much for welcoming me today, and I'm sorry I cannot be there in person and have to be supporting online. So it's a great pleasure to go through some of the best practices for adaptive sampling, provide an introduction, and then move forward onto the bed file design workshop component. So I'm first going to give a bit of an introduction about adaptive sampling, some of the experimental design parameters to consider when preparing for your first adaptive sampling experiments, considerations for library preparation, and then move into MINOW, the software running the sequencing devices, to show how to set up an adaptive sampling run. I'll next go through some monitoring and troubleshooting recommendations, and then we'll move into our bed file design, generation and testing workshop. If you have any questions throughout the seminar and workshop this afternoon, please do not hesitate to ask. So getting right into it, what is adaptive sampling and why use it in your work? To understand adaptive sampling, we first need to understand a little bit about how nanopore sequencing works. So nanopores are embedded into an electrically resistant membrane, and during sequencing, we apply a voltage across that membrane, which creates a flow of current through the nanopores and pulls the DNA across the pore. The passage of DNA is very tightly controlled by this motor enzyme, and it slows it down, or it unwinds the double helix and slows it down to about 400 bases per second. And at this rate, different nucleotides within the pore create a characteristic disruption to the ionic current, which is recorded in the form of a squiggle, which is analyzed and converted into its nucleotide sequence in real time. Adaptive sampling can use this real-time sequencing in order to enrich or deplete regions of interest during the sequencing run. And this is done by comparing the very start of each read to a region of interest. When setting up your run, you upload a reference file into MINOW, and this defines the sequences to either enrich or deplete. When a strand approaches the nanopore and starts sequencing, the first second or so of sequencing is analyzed, base called, and aligned back to that reference in real time, allowing a decision to be made as to whether to reject or accept the strand. So if the sequence, as shown at the top here, is not the region of interest, the polarity across the membrane is reversed and that strand is rejected back out of the pore to make that pore available for sequencing other strands. If the strand does align to the region of interest, it's allowed to continue sequencing. And so this on-flow cell enrichment is something that only nanopore sequencing can achieve. The setup in Minnow software is very simple, comprising a toggle switch, shown here, and then fields to upload a reference and an optional but highly recommended bed file, which we will touch on more this afternoon. So why use adaptive sampling? And the real answer to this is in order to get high coverage of your regions of interest. If we think about a MinION flow cell, for example, using standard untargeted sequencing, you might expect maybe, say, 6-fold coverage for human whole genome sequencing. However, using adaptive sampling, Kovaka et al. Achieved 30x coverage for a cancer panel of 148 genes. Payne et al. Achieved 32x coverage for a gene exome panel of 170 genes. Internally, we've also shown benefit for the analysis of COVID-19 host response panel enrichment for BRCA targets and targeting the major histocompatibility complex. In this graph down here, you can see that with adaptive sampling turned on in red, we see much higher depth for the MHC region of interest compared to the whole genome sequencing in purple. And so the first reason to choose adaptive sampling as your method of targeted sequencing is that because the enrichment takes place during sequencing, it allows for very fast and simple library preparation without the need to optimize primers, probes, additional kits. It makes it really simple to enrich a really high number of targets. The second key advantage is that it is a PCR-free approach. And so this reduces limitations on the read length that are attainable. It allows base modifications to be studied, and it makes it possible to enrich for targets such as repetitive and GC-rich regions that are not amenable to PCR. So once you have decided to use adaptive sampling, what do you need to consider when designing your experiment? Targeted sequences are defined by a reference FASTA file and optionally a bed file that specifies the coordinates within the reference to enrich or deplete. And so I can probably explain this a little bit better with an example. If, for example, you wanted to sequence a panel of 150 genes from human samples, there are two ways that you could do this with adaptive sampling. Firstly, you could simply upload a reference with just those, the sequences of those 150 genes and use just that reference for your adaptive sampling. Alternatively, and the recommended method, is to upload a reference of the entire human genome sequence and then upload a bed file that specifies the coordinates of those 150 genes. And so as best practice, we recommend always having as complete a reference as possible that includes all of the sequences that will be found in the sample reflected in that reference, and then using the bed to specify which regions to enrich or deplete. The reason that we recommend this is because having a complete reference improves the specificity of the alignment, and so you're less likely to enrich off-target regions that are similar to your targets. Once you upload your FASTA and bed file and you start the adaptive sampling, how does the algorithm actually make the decisions? And so what we are looking at here is a schematic of a FASTA reference with regions of interest that would be specified in the bed, shown in yellow. When a read starts being sequenced, an initial chunk of data, so that first second worth of sequencing data that we were talking about, this is typically around 400 base pairs or a bit less, is read, base called, and aligned to the reference in real time. And that information is then used to make a decision as to whether to reject or to accept that strand. So in case A, we can see that the adaptive sampling chunk, so that first second or so worth of sequencing data shown in this reddy brown color here, is located within the region of interest. So this strand is accepted and it's sequenced completely. Whereas in case B, that adaptive sampling chunk is outside the region of interest. So this strand would be rejected from the pore without further sequencing. And so you can see that we run into a bit of a complication here. This strand is being rejected from the pore. However, we actually would have wanted this strand because it continues into this region of interest here. And so this brings us to the first consideration in experimental design, which is the addition of a buffer sequence. And this is a region on either side of your targets that is included in the bed file, so that if your adaptive sampling chunk falls just outside your target, the strand is still accepted. So now we see the situation that we want. In case A, the adaptive sampling chunk is inside the target, and it is accepted for sequencing, giving us coverage that we want. In case B, the adaptive sampling chunk is outside the actual target itself, but it is inside the buffer region shown in red. And so because it's within the buffered region, it is accepted and it's sequenced into that target, giving us coverage that we want. In case C, the adaptive sampling chunk is outside the target, it's outside the buffer, the strand would be rejected, which is exactly what we want because it doesn't have any coverage of the region of interest. And so the next question is, how big a buffer do you need? So if it's too short, as shown in case B, then an adaptive sampling chunk might be outside the buffer, even though it would sequence into our region of interest, and so it's rejected when we don't want it to be. Whereas if the buffer's too long, then strands are accepted when they're not going to give us any coverage of the region of interest. And so the buffer size that is going to be ideal for your adaptive sampling experiments is dependent on the library fragment length, where if you have a short library, a smaller buffer is appropriate, whereas with a longer library, a larger buffer is appropriate. When we think about measuring the length of our library, we often talk about N50. And that's shown in this line here for this distribution of a G-tubed sheared library. The N50 equates to the length of the shortest read in the group of longest sequences that together represent at least 50% of the nucleotides. Similarly, you can have N25, N10, N1, describing the shortest read in the group of longest sequences that together represent at least 25%, 10%, or 1% of nucleotides. So our recommendation for adaptive sampling is to target the library N10 for your buffer size. So if you've sequenced the same or a similar library previously, you can look at the N10 and use this value for your buffer size. However, that's not always possible. And so as additional frames of reference, we have seen really good results with a 10 kilobase buffer for a library targeting an N50 of 5 kb. And that's very similar to what's shown here. Or alternatively, if you're targeting longer fragment lengths, say a 10 kilobase library, we would recommend a 20 kilobase buffer. However, I should mention that there are some caveats to this guidance. And one important constraint for adaptive sampling is that that we recommend that the enriched portion does not exceed 10% of the total sample. And so this is quite a busy slide, but the key takeaway here is that in example 2, where there are a very high number of targets, adding a long buffer that equates to the N10 of that library would mean that the adaptive sampling would be targeting over 15% of the entire human genome sequence. And so we would not see as good enrichment as we would with a shorter buffer. And so, yeah, it is worth keeping this in mind in addition to the guidance on using an N10 as your buffer size. One way that you can reduce the total amount of on-target library, if you're pushing this 10%, is to use a directional buffer. So the idea here is that if an adaptive sampling chunk is in your buffer, but the sequencing direction is away from the region of interest, as is the case for B and also for D, there's no value to accepting that strand. And so it is possible to add a directionally aware buffer so that your region of interest is buffered upstream for the plus strand and downstream for the minus strand, so that you're only accepting these reads that are heading towards your region of interest. And when we move to the bed file design workshop, I will show you this directional buffering. It lies in putting the directionality in that 6th column of your bed file, and we will look at this more later. I mentioned earlier that it is best practice to, wherever possible, use a full reference that represents everything in your sample, plus a bed file for targeting your regions of interest. So for a human genome example, uploading the full human genome reference FASTA, and then specifying your regions of interest in the bed file. However, there are some computational limitations to this. So on a GridION, P2 integrated, P24, P48, FASTA files up to 10 gigabases will run smoothly. And so a full reference is going to be possible for most genomes. The Mark 1C is very different to this. So there is a maximum reference size of 125 megabases. So if you're using Mark 1C for enriching larger genomes, the reference FASTA will have to be just a subset containing only the sequences of interest to be enriched plus their buffer regions. And you also need the extra step to take care that this does not exceed 125 megabases. So hopefully you now have a better understanding of the theory behind your reference file and bed file design. How do you go about sourcing or generating these files? This will be the focus of the second section this afternoon. However, to cut to the chase, we recommend downloading reference files from trustworthy databases such as the UCSC Genome Browser, which is what we're going to be using later this afternoon, NCBI RefSeq database. And in terms of your bed file, we also recommend double checking that it's free from formatting errors and correctly matching your reference using the Bedbug's Bed Checker tool. And again, we will go through this afternoon. Finally, we do also have an adaptive sampling catalogue, which is a location where Oxford Nanopore and community members can share bed files. And so some commonly used ones include the hereditary cancer gene panel and reduced representation methylation sequencing bed files that capture CPG island sites. So once you've planned your experiment, you've sourced your reference and bed files, it's time to prepare your samples and libraries. And so I'm going to briefly take you through some of the considerations for fragment length library preparation and library loading. Adaptive sampling is compatible with a really wide range of fragment lengths, but you will likely see the best coverage of your targets with shared libraries, shared to between 5 and 12 kilobases. And there are a few reasons for this. The first is that sharing reduces pore blocking, and that's what we're looking at on the left. So you can see a 6 kb library and a 20 kb library. And for the 20 kb library, we see that pores are being lost to this teal unavailable state that's characteristic of terminally blocked pores. And this reduces output, and it will necessitate the recovery of these pores with a flow cell wash. So because of this faster blocking with the longer libraries, you will get better coverage of your regions of interest with a shorter library. The second reason that we recommend sharing libraries is that it can increase enrichment depending on the size of the individual regions of interest. So let's say, for example, your targets are in the 2 to 5 kilobase range, but you have 30 kilobase fragments. You're going to be sequencing a lot that is off target simply to get coverage over your region of interest. And so aligning your library fragment length to your on-target region of interest length is also helpful. Finally, shearing helps facilitate molarity calculations, and this is important. I'll talk about it in just a moment. We have validated methods for sample shearing available from our documentation tab on the community, and there are methods for high throughput genomic DNA shearing using GenoGrinder or FastPrep 96 for GTube and for MegaRaptor 3. I should have mentioned, I've talked about why we don't, or why you'll get better coverage if you avoid too long a library. But it's also important to note that if you have a very short library, so say cDNA library, adaptive sampling is not going to be very effective either. So this 5 kilobase is really the lower end of what you would be looking for in your library in order to get good enrichment. And the rationale for that is that if you are rejecting a read after, say, 800 bases, but it's only 1 kilobase long, you're getting minimal benefit from having rejected it. It only saves a very small amount of time, and so you do not get good on-target enrichment if your library is too short. High pore occupancy is critical to maximizing output across all sequencing experiments. But because adaptive sampling is constantly rejecting strands, having high pore occupancy is especially important for adaptive sampling runs. So when I talk about pore occupancy, I'm talking about the percentage of available pores that are actively sequencing rather than open and available for sequencing, but without any strand in them at a given time. And so we can look at this in Minnow as the light green sequencing pores as a percentage of the total, light green and dark green, where those dark green pores are available but not sequencing. So here we're looking at the pore occupancy of an adaptive sample, of 2 adaptive sampling runs, and we can see that the occupancy keeps improving up to 100 femtomole. And so for adaptive sampling runs, we recommend loading at least 100 femtomole for best results. And this is higher than our typical recommendation for most kits. In order to achieve this high pore occupancy, we recommend ligation-based kits for adaptive sampling. And so for single-plex, you would be looking to use SQK LSK114. For multiplexing, you'd be looking to use NBD 114.24. If you are wanting to multiplex multiple samples onto a single flow cell, you can pool your samples into one library as per typical multiplexing. However, you do need to be wary that barcode balancing can be challenging and that you may end up with higher coverage for some barcodes than for others. And so the alternative here, if you're really trying to maximize the on-target output for each of your samples, and get the most out of your flow cell, the alternative would be to barcode your samples individually and then run them sequentially, washing the flow cell between samples. Finally, to get the best depth of coverage, we recommend washing the flow cell where appropriate to recover terminally blocked pores. And so as rough guidance, when only about 20% of the remaining pores are available, it would be worthwhile washing. And so for this, we're looking at the green as a percentage of the green pore available and teal unavailable. And so you can see the washes denoted by an asterisk up here. Those washes recover unavailable pores, allowing more to be available for sequencing. For many adaptive sampling runs, it's appropriate to wash after about 24 hours, but this is library dependent. And as in this example here, I would look more to the plots and the pore availability than focusing on the clock. So with the recommendation to load 100 femtomol and the recommendation to wash and reload your library, depending on your kit and multiplexing setup, you may require multiple library preps in order to achieve best results from adaptive sampling. So it's good to be prepared for this up front. Okay, we've gone through reference embed and best practices for the library. I'm now going to take you through the actual setup within Minnow. Before I get into the software, I just want to explain by way of background that adaptive sampling base calling and live base calling are two independent base callers. The same goes for the adaptive sampling alignment and the live alignment feature in Minnow. So adaptive sampling uses a fast base caller and an aligner optimized for short reads. And both of these are turned on when adaptive sampling is selected. The user has no control over them, does not see their output. Alongside the adaptive sampling inbuilt base caller and aligner, it is possible to turn on or turn off live base calling and live alignment. Because these two processes run in parallel, they compete for resources. And so if you have too high a model of live base calling selected, or if alignment is using up too much of your computational resources, then that can slow down the adaptive sampling decision. And so it's just important to be aware that the settings that you choose for your live base calling and alignment are not impairing your adaptive sampling. This will depend on your computer setup. So within Minnow, setting up an experiment is very simple, and it's very similar to setting up any sequencing run. So you just click Start Experiment, input an experiment name, and then click Continue. Choose your sequencing kit and continue. You then use this toggle button to turn on adaptive sampling. You'll get a pop-up where you input your reference, FASTA file, and your bed file. Advanced options allow you to only perform adaptive sampling on a subset of channels. You can choose enrich or deplete and click OK, then review and click Start in order to start your sequencing run. Once you've started your run, it's good practice to monitor it and ensure it is running as expected. One of the key variables that we think about is full loop response time. The full loop response time is the time taken from a strand beginning sequencing to being rejected from the pore. And so this includes about 1 second of data acquisition, like actual sequencing time, between 0.35 and 1 seconds of data handling time, for base calling, alignment, and comparison to the bed file. And on a PromethION 24 or 48, an additional 0.4 seconds for the unblocking process that rejects the strand from the pore. Now, the longer the full loop response time, the more time that is spent sequencing off-target reads. And so your full loop response time directly impacts your enrichment. A 15% increase in full loop response time can reduce your on-target output by about 10%. And so it's important to monitor this. But how do you actually do that? And the way that we look at full loop response time is by monitoring the rejection peak. In every adaptive sampling run, we expect a major peak in the read length histogram that corresponds to reads that are rejected from the pore. And so by looking at the length of these reads, we can gauge the full loop response time. And so to view this, we're looking at read length histogram using the scale bar to zoom in to that shorter read length in order to look specifically at that rejection peak. Turn on split by read and reason to view the adaptive sampling voltage reversal peaks. That's our rejection peak in purple. And if you're running live base calling, select base called in order to see more accurate data in terms of read lengths. And so what we're seeing here are expected rejection peaks for GridION and PromethION runs, and variation greater than about 15% of typical should be seen as indicative of an issue with the run. And that could be GPU performance, it could be dedicating too many resources to your live base calling or alignment, or it could be a quality issue where due to low quality, it is taking too long for the decision making to happen. You're also looking for reads in green showing up at higher read length. And so these are your accepted reads. This is often going to be a very small proportion of your sample, and so it will typically look very small. But again, you can use the scale bars to slide to avoid seeing that rejection peak, and this will rescale the y-axis to better show you your acceptance peak. As I mentioned, if you are seeing a delay in decision making, make sure that the device does not have other processes that are occupying resources. Make sure that you're complying with the limits of performance for each device. So this is ensuring that you're not using live super accuracy based calling, for example, not using high accuracy unless you have a GPU that can support it, turning alignment off if required, etc. And then also checking for sample issues, things like low Q-score, incorrect translocation speed, device temperature, can cause quality issues that can also delay decision time. One really useful file for troubleshooting and evaluating your adaptive sampling runs is the adaptive sampling CSV in the other reports folder of your sequencing output. And so this lists the read ID. The sequence length in this file is the number of bases that the adaptive sampling script used to make a decision. And then it also lists what that decision was, where unblock means that the strand was rejected from the pore, it did not align to the bed. Stop receiving means that the strand was aligned to your target region and it was sequenced to completion. Unblock hits outside bed means it was too small to align, and no decision means that a decision could not be made. So you'll be mostly seeing unblock and stop receiving. Importantly, the percentage of stop receiving should correspond to the probability of capturing a strand of interest, which in turn translates to the percentage of strands accepted by the adaptive sampling script. So as an example, if your bed file specifies 2% of your genome as on target, you would expect approximately 2% of reads to have the decision stop receiving. And any major deviation from this suggests that the decisions are not reflecting what you expect and you need to take a closer look. So the take-home messages from this introduction are to make sure that your input DNA is of good quality. Use ligation sequencing, so LSK114 for a single sample and BD114.24 for multiplexing. Shear your DNA and ensure that your fragment length is between the 5 and 12 kb range. Load 100 femtomole of library onto your flow cell. Use the wash kit when a large proportion of your pores become unavailable. Whenever possible, use a reference representing the entire sample and then specify your target region with a bed file. Use a buffer length corresponding to the N10 of the library read length. And again, as a ballpark, for a 5 KB library, consider a 10 KB buffer or a 20 KB buffer for a 10 KB library. Ensure that your minnow settings are not causing too much competition between your live base calling and or alignment and your adaptive sampling. And so monitor your rejection peak to make sure that you're not overstressing your computational resources. And as a good first recommendation, we would typically use fast mode for live base calling with live alignment off for most setups. With that, I'm going to move over to the bed file construction part of today. Just a moment. So hopefully you're now seeing the documents sent around to be used for the workshop. So what I'm going to do is take you through an example of how to generate, modify, and buffer a bed file that could be used for adaptive sampling. We'll be using as an example a hereditary cancer panel. The method used for this bed file construction is only one method available to you. Ultimately, anything that results in a valid bed file is going to work. I have designed this to use, rather than command line tools, as many point and click graphical user interface tools as available. And so I'm going to be using an internet browser, a text editor that has end-of-line conversion capability, and Microsoft Excel to step through some of the modifications to the bed file that we will be performing. I will jump over into our genome browser. Okay. Before I continue, could I please check that everyone is able to get out their computers and follow along for this component of the workshop, that you have received the instructions in the PDF, and that you have available for you the Sublime Text Genome Editor.
Bapak Ibu, apakah datanya sudah siap dan browsernya sudah siap? Jika ada yang masih belum mendapat akses dari PDF, boleh angkat tangan. Tim kita akan langsung datangi Bapak Ibu dan membantu Bapak Ibu sekalian. Begitu juga untuk Sublime Text-nya. Jika ada yang masih berkendala, boleh angkat tangan. Tim kami akan segera bantu. So Rebecca, our team is currently checking with the participants. So if it is okay with you, we We are going to wait for several minutes. Thank you.
No worries.
Hi, Rebecca. Can you hear us? Yeah.
I can, yes.
Okay, thanks. So while waiting for the setup, I have one question that hopefully will stimulate maybe discussions. So you've talked about 10% sizes for the target region of interest. Is there any minimum size just to get us started?
Look, we currently do not recommend any specific minimum. The lower the percentage on target, often the better the enrichment you will see. However, there's, so if you're targeting, say, 1% of the genome, you'll often see better enrichment than you might at 10% of the genome. However, as you go too far below that, you're not then getting the benefit of the adaptive sampling so much. So if you're really only interested in one gene, you can do that. And our current recommendations are that there is no real minimum. However, you're likely to see very similar enrichment if you were to do, say, 20 or 100 genes. And so my personal thought is that you might as well pick some additional targets, things that could potentially be of interest, and include those, because including them isn't going to have a detrimental effect on your enrichment of your true target. So I hope that makes sense. And short answer is no minimum.
So by more better enrichment for like around the 1% sizes, do you mean that more coverage or better coverage?
Yes, yes.
Right, so yeah, better coverage or better enrichment if you target smaller parts of the region around 1%, but you can go up to 10% for adaptive sampling.
Yes.
All right, thank
yes.
you.
Okay, while waiting for the others, is there any questions that you want to ask to Rebecca? Okay.
Okay, thank you, Rebecca, for your presentations. My first question is, should we prepare the DNA with the high molecular weight DNA kit extractions, or we can just use any standard of DNA kit extractions? And the second, it's interesting to see that you are mentions that with the MEC1C, we can use that the adaptive sampling below 125 megabase pair, while for the grid ions, it's can more data that we can use. Is that because the grid ion has the 5 flow cells, means that we need to multiply the flow cells that we use for that one, or it's because another reason of that? And the third question is, since you in ONT also offer about the enrichment and also the depletions, and probably you have some best practices, especially because when we talk about the ribosomal RNA is something that probably will be very abundant. So in any cases, is there any considerations whether we need to put the depletions or the enrichments, considering we have kind of also considering that we have different type of organisms. Also, especially in human, also we have a lot of the transposable element within the genome. And yes, probably that's questions. I will follow up next. Okay, thank you.
Thank you very much for the questions. Let me work backwards because I'm remembering your most recent question better. So in terms of enrichment or depletion, you can only do one at a time. If you are interested in specific targets and you know the sequence of those targets, use enrichment. So if you can, use enrichment. The cases where you would use depletion would be more in situations where you do not know the specific target that you want. And so, for example, if you have clinical samples and you are trying to understand what is the infectious agent, what is the pathogenic organism causing an infection, you do not know what that sequence is going to be, but you do know that in your sequencing, you're going to have a lot of host, and you're not interested in the host, you're interested in everything else in order to attempt to find the infectious agent. That's the kind of situation where you would do depletion. So you would upload the human genome as your depletion target. For GridION versus Mark 1C, the sizes that I was talking about are actually the sizes of the reference FASTA file that you can use. So it's not so much about the amount of data that you'll get back per flow cell. It's really a computer limitation. And so the computer in the Mark 1C does not have the memory to use an entire human reference genome as its reference. The reference needs to be less than 125 megabases. And so it's not really flow cell related. It's not really output related. It's really just the size of the reference that the onboard computer can cope with. And then, I'm sorry, could you repeat your first question?
Weight DNA kit extractions, or we can just use the...
Yes. It doesn't need to be particularly high molecular weight. You're targeting 5 to 12 kb. I suppose the caveat to that is if you are trying to phase long regions, then you need high molecular weight DNA. And so you can use adaptive sampling for those kinds of applications. It will work, it will block faster, but you will still get enrichment and higher coverage than you would without adaptive sampling. But if you're using it for, I suppose, fairly standard applications, you're targeting, say, a 5 kb library, you don't need a particularly high molecular weight extraction kit. And I would encourage you to go to the Nanopore community and look to see whether there are validated extraction methods available for your sample type and use that as initial guidance. But yes, it doesn't inherently need to be particularly high molecular weight.
Okay, so probably I will add other questions. So using the calculations for the adaptive samplings, do we need also to calculate the coverage estimations or for example, from 100 genes or something like that, or we just need to calculate the fractions of the genes with the genome? because sometimes, like usually, like for example, for amplicon sequencing, we try to see for 100 or 200 of times of coverage, but with this kind of approach for adaptive sampling, do we need to also calculate the coverage in the end, or only the fractions of the genes within the genome?
That depends on your sequencing goals. Often you will want to know what your coverage is. I mean, for a lot of applications, especially for human genome sequencing, for the example that we're doing today, targeting hereditary cancer genes, you'd probably be looking at, say, around the 30x depth, as in order to give you good variant calling. But that's really your sequencing application and the requirements of that, rather than anything specific to adaptive sampling.
Okay, thank you, Rebecca.
You're very welcome. In the interest of time, shall we move forward with the workshop component? We only have about 10 minutes left for this session.
Okay, ladies and gentlemen, can we move forward, or shall we wait for a little bit? Or is there any questions regarding this workshop? Okay, Rebecca, I guess we have to wait for another 2 minutes. Is it okay?
No worries. We may not, I don't know how tight on time we are at your end, so I'll be guided by you as to when to move forward and when we need to stop.
Okay, sure. Okay, Rebecca, currently we are downloading the data, but I guess we can move forward to the next step. So please go ahead.
Great, yes, fantastic. So I wanted to start just on the UCSC Genome Browser homepage. And so the first step in the PDF is to download the data. Fantastic that you have started doing that. If you're trying to navigate there from the main homepage, go Downloads, Genome Data, Human Genomes. We're looking for the HG38, Standard Genome. Scroll down, and then it's just the zipped hg38.fa.gz file that we are downloading. While that's downloading, we don't have to wait for that to finish, but we can continue through to the table browser. So this table browser is really a really handy way to input a list of gene symbols and to get back a bed file that we can use as a preliminary file for developing the bed for adaptive sampling. So at the bottom of the PDF, there's a list of gene targets. I'm going to copy those. And then over in the table browser, what we're going to do is we're going to check that we have the HG38 assembly. So this is the assembly that we've just downloaded from UCSC Genome. So we want to make sure that we're creating a bed file that matches that. For the track, we're selecting NCBI RefSeq. And for the table, we are selecting RefSeq Select and Main. This table means that we're only going to get back a limited number of entries per gene symbol that we input. So if we were to choose, say, NCBI RefSeq All, then we would get back a lot of different entries for each of a lot of different rows in our bed file for each of the targets. And so by selecting just the main, we're limiting that to, in most cases, just one per target. We'll then paste our list of genes using paste list and click submit. Then from the output format, we'll select bed. I'll leave this open for a moment. Let me know when you're ready and everyone has selected these options and pasted the list of gene symbols from our PDF into that input box. How is everyone going with this step?
Okay, Rebecca, we are currently waiting for the steps. I guess we are going to wait until 3 minutes more.
Great.
Okay, Rebecca, we can move forward.
Fantastic. So once you have, Ref 6 selected main, you pasted your list in and you've chosen bed as the output format. Click Get Output. Create one bed record per whole gene is correct. Click Get Bed. And you should receive this. So this is a bed file. The parameters or the columns that are required for adaptive sampling are columns 1, 2, and 3. And the rest are somewhat optional for adaptive sampling. Column 1 is the chromosome reference. Column 2 is the start coordinate of the region of interest, and column 3 is the end coordinate of that region of interest. One thing that we also have in this default, or in this bed file created by the UCSD table viewer, is a directionality. And so I mentioned that this can be useful for buffering, but at the moment, it is not helpful for adaptive sampling because it specifies only one of the strands. So because this specifies for this first gene only the minus strand, if any reads aligned to the plus strand, they would not be accepted for adaptive sampling. And we do want both the plus and the minus strands to be accepted for adaptive sampling. And so we need to remove this directionality information. In order to do that, I find one of the easiest ways to modify the table and select only the information that we want is to put it into Excel. So I'm going to right-click and save as. Please follow along. Right-click, save as. We can just save it as default into the downloads. And then in Excel, I'm going to go to Data from Text or CSV. Go to my downloads and find that HG38 tables that we just saved. It's going to give us a preview of how it's transforming that data. So it's a tab separated file. Excel is automatically detecting that. So we just click load. And this is the information that we want in our bed. The reason that I'm putting it into Excel is to make it really easy to just choose those first 3 columns. So again, this 6th column is providing information that's only specifying one strand, and we don't want that for adaptive sampling, at least at the moment. And so we're just going to select columns 1, 2, and 3. Without selecting the header. So I did not select the header set, but I just selected columns 1, 2, and 3, and I'm going to copy those. I'll pause for a minute to make sure everyone's been able to import this into Excel. Select columns 1 to 3 without the headers and copy them.
Okay, we're gonna wait for 5 minutes. Bapak Ibu setelah dapat data Excel-nya bisa di-copy kolom 1, 2, dan 3 tanpa headers-nya ya. Okay, Rebecca, we can continue.
Fantastic. We're then going to move over into our text editor. I've recommended Sublime Text because it has the ability to convert the end-of-line characters. So if you have Sublime Text, please use it. If you do not, you will likely encounter an error when we check our bed file. But please continue to follow along, see that error, and once you get Sublime Text, you can have a go at converting the end-of-line characters. So what I'm going to do here is I'm just going to paste that information that we just copied from Excel. So we now have a file that has the chromosome reference, start coordinate, and end coordinate for each of our regions of interest that we sourced from the UCSC table browser. You could equally, if you know the coordinates of your genes of interest, you could equally just type this out. It's a tab separated file. So you could equally just open up Sublime Text, type in your chromosome reference, type in the start coordinate tab, type in the end coordinate, and that is an equally valid way of doing it. I'm going to remove this last line that we don't need. Scrolling up, I'm double checking that we do not have any headers. Now, at the end of each line, the enter character is different for Windows, Linux, and Mac. And this can cause problems when it comes to creating your bed file. The correct formatting for the bed files for adaptive sampling uses Linux line endings. And so the reason that I chose Sublime Text is that I'm able to view line endings and convert these into the Unix format. So if you're in Sublime Text, please follow along, view line endings, and choose Unix. Once we've converted the line endings, I'm going to save this as hereditary cancer panel.bed. The save as file type, just choose all files, and we're inputting this bed file extension to be used for this file. I'll pause here for a moment to make sure everyone can catch up.
Okay, Rebecca, we can continue.
Great. I'll save that. We're now going to check our bed file. So Oxford Nanopore developed the Bedbugs Adaptive Sampling Bed Checker, and so you upload your bed file and upload your FASTA reference file, and then this will check it for formatting errors. So I'm going to drag in our bed file. I'm also going to drag in our downloaded reference FASTA file. It's still unzipped, and that's fine for checking. One thing that's important is that there are no spaces in your FASTA reference file. It will cause problems if there are. So here there is a space. I'm just going to rename it. If it's the first time you've downloaded this, you won't have the one, and so it should be automatically just hg38.fa, but I wanted to leave it to make sure that I made the point that you avoid spaces in your reference files. And so I will then drag that on as well. So you've got your bed file and your reference FASTA file uploaded, and we're going to begin analysis. So it will take some time to analyze the bed, cross-reference it to the FAST-A, and then it will give us a summary. And so we see this validation report. And so what we're looking for, if everything has gone well, we see sea of green. If you have not converted your end-of-line characters, so if you're still using Windows or Mac characters for the end of line, this box will show up in red. If you have a space in your reference file name, or if your bed and your reference do not match, the reference overlap will show up as red. And so one reason that this can happen is if you download your reference from NCBI, but you use the UCSC table browser for your bed file creation, the chromosome names will not match. So NCBI uses different chromosome names. It doesn't name the chromosomes CHR1, for example. It uses different names for the chromosomes, and so it would not match, and this would show up red. Self-overlap just means that some of the regions in the bed file overlap with each other. This is not inherently a problem, and adaptive sampling can proceed even if this is red. It also gives us a total number of nucleotides and a total interval count. So we're targeting 138 regions of interest with a total of 11.8 megabases. And so we can work out the percentage of the genome that we're targeting. So we've got 11.8 megabases divided by 3.2 gigabases, which is 3,200 in megabases, or multiply it by 100 to get a percentage. And so we're targeting 0.36% of our genome. And so it's a nice, easy way to see that. So this is fantastic. Our bed file has been created. However, it has not been buffered. So we do not yet have a buffer. One of the handy things that the bed checker does, in addition to making sure that formatting is correct, is it allows you to download an extended bed file. And this is a directionally aware bed file. So I'm going to click there, save this. And then I will open that in here. So this was our original that we uploaded into Bedbugs, and this is what it has returned. And so what we can see is that for each entry, there are now two copies, one on the plus strand and one on the minus strand. So we are selecting for both strands, which we want. And the buffer has been added upstream of the plus strand and downstream of the minus strand. So you can see that it's 24148 for the plus strand, but 24149 for the minus strand. So we've added a 10 kilobase directionally aware buffer into this bed file. And so this bed file is ready to be used for adaptive sampling with a library shared to an N50 of 5 KB. That concludes the workshop component. We have probably less than a minute, but please do ask any questions if any issues arose while following along.
Okay, ladies and gentlemen, we've come to the end of Rebecca's presentations. We open one questions for this workshop. Is there any questions? Okay, I guess we are still wrapping up the trial here, Rebecca. So therefore, I guess we are going to continue afterwards, and I guess this is the end of our sessions. Thank you so much, Rebecca, and let's give a round of applause to Rebecca. Thank you so much. Now
Thank you, everyone.
Yeah. Okay. Now we are going to move to our second facilitator, which is Dr. Pirun, that will facilitate us to Epi2Me Lab's human structural variation analysis. So Dr. Pirun is a distinguished bioinformatician with over 15 years of expertise in next-generation sequencing data analysis. He served as an instructor at the Division of Medical Bioinformatics, Faculty of Medicine, Sri Raj Hospital, Mahidol University, and is an adjunct instructor at the University of Arkansas for medical science. With a Master of Science and PhD in Bioinformatics, Dr. Pirun has conducted a groundbreaking research in third-generation sequencing with more than 30 peer-reviewed publications. His current focus is on developing bioinformatics tools and pipelines for human genomics and pathogen research. So for Dr. Pirun, we are invited you to the stage, and let's give a round of applause for Dr. Pirun.
Hello? It's working. Okay. Good afternoon, everyone. So this is, okay, my slide is already there. All right. This part is going to be how we can analyze the data after we get the adaptive sampling just now. So Rebecca already showed you how to build the bed file. And actually that is the beginning step before you do sequencing run, right? And then when you want to do sequencing, so you prepare the DNA. Let's go to my slide here. So you have the cell, you extract the DNA, right? Then you prepare the DNA with the adapter. So before you loading before you running the sequencing, so you have to select whether you want to do the whole genome sequencing or you want to do adaptive sampling. So, and with that, you can select adaptive sampling if you want, and then you provide the bed file that you have created just now into the Epi2Me, into the Mino software, and then run the sequencing. So after that, you're gonna get the data, right? So sequencing go to the pore, and the pore change the signal into the ATCG-based DNA. And then that call base calling. And then base calling, you check the quality, whether the data is good, the read is good enough. So if it's good enough, so you go in the data analysis process. And the whole things here, the QC step and data analysis will handle into the Epi2Me that you can use very easily right now today. So the Epi2Me desktop is actually the new release. Previously, Nanopore have the Epi2Me cloud or so-called Epi2Me portal that you can upload the FastQ to the cloud and then analyze the data on the cloud. But right now, this one, the Epi2Me desktop that you have already installed in your computer is the local version that you can run the Epi2Me on the laptop or on the desktop that you have. And in the future, I think they're going to have the cloud version, but right now it's not released yet. So that EP2Me is user-friendly, so very easy to use, and the person who have just limit skill of the bioinformatics, so you still can do it. But the process of the installation is a little bit difficult for us, so it would be the good idea if you have IT or bioinformatician with you when you do installation. So, and this one, the software actually can run either Windows, Mac, Linux, doesn't matter. You can download and install on that one. Okay, so I know that most of you have the VM that you can use the computer on the cloud, right? They have the VM access for you. And some of you already installed EP2Me on your laptop. So I provide the instruction, so I think you can go through and then do the installation on that. So, but this is the page that you're gonna go, so it will be the installation page, and you can click on the download here. It's gonna show you this page. You can select either the platform that your computer belong to, Windows 10 or 11, so you use that one. So Macs, they have two versions of the Macs. So if you buy the Mac OS like 3 years ago or 4 years ago, so it's probably Intel version. So you need to download that one. But if you just buy the new Mac, so it's gonna be the M1 or M2, M3, M4. So that should be this version that you have to download. Ubuntu, yeah, this one, and the rest is this one. Okay, so, and yeah, as I said, when you're going to install, so please make sure that the IT team or bioinformatician with you, so to make it smooth in terms of the installation. The difficult part is actually these two guys, Nextflow and Docker. So that the EP2Me actually need these two software to install into your computer. Yeah, so and the configuration, some computer is very easy, like MacBook Silicon, so that quite straightforward. For Windows, maybe some technical problem with the WSL, Windows Subsystem Linux. So then you need IT or bioinformatician to help you out. So yeah, install with them, that's the basic. And then after you install, you're gonna have the software on your computer. In my case, here, I'm gonna have the EP2Me over here. So once I open it, it should be go to in this screen. This is my EP2Me on this computer. So, and the first thing that you will check is whether your setting is okay or not. So you go to the setup local, and then you check whether Nextflow is good. If it's busy and it is in the red color, so it means that you have problem. So therefore you need to contact your IT or bioinformatician. And then after this one is ready, so you go to the next, continue, is gonna show you the Docker one. And if your Docker is ready, so it means that your EP2Me is ready to use. Okay, so and click finish. So, and the step is over here actually. If you go to and check the step, go to the workflow here, step 1 is here, the setting. And then step 2, upload local workflow, click and check again Docker, everything fine, you go to the next one. So now, EP2Me is installed on your computer. Next, you have to install the pipeline or workflow that you want to use in your analysis. So in this session, so we're gonna install WF human variation pipeline that you can analyze the data of the human genome, whole genome sequencing or adaptive sampling of the human genome. So to install that, again, very easy. After EB2Me, everything easy. So before, it's a bit difficult. Anyway, when you want to check what is the available workflow that you can download and install into your system, so you go to this one, available workflow. After you click, you will have the list of the workflow that you can use in your analysis. So it has a lot of workflow, like workflow alignment, workflow human variation, workflow metagenomics, workflow 16S is all here. So you can select the workflow that you want to install. So, and in this case, we want to install human variation, so we click on the workflow human variation, and then it's going to ask you that whether your computer is powerful enough to run the human variation workflow. And this computer, my computer, is not enough actually, but I install anyway. So for your research, if you want to analyze your human variation information, so please make sure that you have enough CPU, should be like 32 CPU at least, and then the RAM should be 128 gigs or more. So that the requirement that you can run for the whole genome. If you have just some region of the human, like adaptive sampling, you can have lower setting. So, and that you have to change and check whether it's enough to run or not. So, yeah, so then this is the minimum that you can go. CPU 12 and then RAM 32. If you don't have enough, then it may have some problem during the run. Okay, if everything is okay, then you click install. And in case of technical in Windows or in Mac, sometimes Docker not allow you to use the whole disk space and the whole environment information. So you need to go into the Docker itself and then increase the space and also the RAM and CPU. So depends on what they need. So previously I run on my computer and it's not allowed to get the space up to 32 gigs of the space. Therefore it's failed during the run. So therefore I go to the Docker and then select this one to increase it. The default of my computer is 8 gigs, so I increase it to 32 as the minimum request of the pipeline. So, and then so actually, if you want to go to the Docker, it should be over here, dashboard. And then you have the setting to change the resource over here, and then change, just increase a little bit. By default, it will be set at 8 gigabyte. So you can increase up to 32 or more. So depends on what you need. And then you save and restart. So then the Docker will restart again. So that in case you have the problem. All right. So, and actually Nanopore already have the way how you use this pipeline. So if you go to the Nanopore community, and they have the tab so-called Nanopore Learning, and then you can see they will have the video of the EB2Me lab human variation pipeline. So you can visit that site as well. It will be very similar to what I showed you today. So after you install finish, then it's gonna present in your list of workflow how you can check that. You can check on the installed here. And after you click that, it's gonna show the pipeline that already installed for you. So in this case, we only have the human variation pipeline, so it will be shown here. Okay, so, and this is the real one. So if you click on over here, workflow, Then you click on the installation, you have this one as the workflow. Here the available pipeline, here the install one, okay? All right, so if you click next, now the time to run the human validation pipeline. So to run it, first you go to the pipeline itself, go to this workflow, and then go to the installed, and then click on the workflow human validation. After you click, you're gonna have this all information before you run, okay? The first thing that you can check, what is this workflow do? So you can click over here. So this is for more information that if you need more explanation, after you click, you're gonna go to the pipeline itself from the GitHub. And then this will be show you all the detail regarding to the information about this pipeline, like you want to identify structural variation, single nucleotide variation, copy number variation, STR, and so on. So, and this more the detailed information of that. Okay, so, and once you understand that pipeline already, so after you read the whole things here, then you gain more confidence how to use it. Next thing is that you have the list of the information here. The first one here is when you're ready to run the pipeline, so you click over there. And right now, I didn't click it yet. So this one, in case you just finish install and then you want to try whether this workflow is working on your computer or not. So you can try the demo data. So you don't need to use your own data. So after you install, you can run the demo data and check whether the demo is successful on your computer. And then update workflow is here. So Nanopore actually quite often update their workflow. So you can check the version over here, and it change maybe 3 months once, every 3 months or 6 months change. So you can check on that. So and they have some update, so you can read the update what they update on the workflow. And then you can switch over between the version. So this one, if you see that the new version have some problem, so you can get back into previous version. So you can select this specific version that you like. And then if you don't need this workflow anymore, so you can delete it over here, okay? And this is the output of the workflow, where's the open the workflow, where's the workflow located on your computer. Okay, so we're gonna run the human validation pipeline right now. So we click this one, run this workflow. And then it's gonna show you this one, okay? So I think everyone can follow me right now. I mean, for your VM Linux, then we can run together. So that start from here, okay, start from here. So you open the Epitome on your computer. So, and if it's done, let me know. Done, really fast. So, and this side, have you opened the Epitome yet? On the right side here, let me know if it's done. Are you open the Epitome yet? Okay. All right, so if you already done that, yeah, you can click over here, run on your computer, okay, and then click continue. Now, they have the setting for you that you can change, depends on the run that you have, okay? So they have many options that you can run, either structural variation, SNP, or single nucleotide variation, copy number variation, STR, short tandem repeat, expansion. So that Dr. Ira present this morning. So this one, they have the list of the genes related to repeat expansion. If you have the new gene, you can add more gene on that pipeline as well. So, and they have more like, so just click this one to show more detail. Okay, continue. Yeah, you have copy number variation, you have modification. So as you know that DNA have, sometimes you have the modification on the DNA itself, like M5MC. So then if you want to identify that 5MC, you can click over here. So, but based on my computer, which is just the laptop and just the demo as well, so I'm gonna click only two things here. So it's the structural variation that we want to find and single nucleotide variation that we want to identify. And if you want more, you can click more later on. So for this workshop, just two things here. And then the sample name. So the sample name, you can change the name up to you. You can change to any name. So in this case, we change the name as the sample we have, like CMK dash what 86. So dash MOD is actually my modify. I add some gene inside. And then the next step is that you need the BAM file. The BAM file is the file that you perform the alignment of your data to the reference genome. So this one actually you can select during the run of the adaptive sampling, but Nanopore might not recommend to align during the run. So it's better to do alignment after the run. I mean, after the sequencing finish, then you perform the sequencing alignment yourself, and then select the folder that have the BAM file. Yeah. So right now we have BAM file over here. So in your case, it will be /home data, right? So you will have the folder name data. And then once you click into the data, you're gonna have this file inside. So then select that data file and then open. And next, it will be the reference. So in this case, I download the reference for you. So this is the reference that human variation pipeline gonna use. So if you perform the alignment to this genome, it's not gonna align again. But if you not align to this genome, so it will perform the alignment during the run. So that depends on what you need. So from my experience, if I alignment with this genome is much better, because sometimes we have pipeline collapse during we use different reference, and then it will do alignment and it's stuck in some step. So therefore, I would recommend that you do alignment with this genome first. Yeah, and then after you align to this genome, then you can select the genome to this pipeline. All right. And this step, the BAM file, the coverage here, the BAM minimum coverage, normally don't change it. Okay. So this is the minimum coverage that you need for confidence of your core. If you have more data, you can increase it, like 30x coverage, like 30, something like that, if you have really high depth data. So, but for the normal run, you can keep 20 is good enough. However, in this run that you have in hand, we control the sequencing depth to make it can run on my computer. So therefore, I decrease this one into 10x, 10 coverage. So this is not recommended for the research, okay? So it's just for demo today. By default, use 20 right? Keep note into your notebook. Yeah, minimum should be 20. And then the last thing here, the bed file. So since you do the adaptive sampling, so therefore the sequencing will be sequence only the location that you amplify, right, or enrichment. So therefore you can select the bed file that you prepare before, like the one that Rebecca showed you. So you select that and then put it here. So it's better to use the bed file before you perform the last step. So the bed file that you download from UCSC, so that one will be the precise location of the gene. Yeah, but this one for the adaptive sampling, so they will add 10K up and 10K down of your region, and then when it's calculated, the coverage it will be a little bit different. So therefore, yeah, choose the one that you download from the UCSC browser. And then annotation is click by default. If you want the facing, you can click facing. So, but right now I keep everything as the default. And then we go to the advanced a little bit more. So sometimes if you run the new run, it can automatically identify the base caller for you. But if not, you can select it. So that depends on how the base calling you use during the perform base calling. In this case, we use version high accurate and version 5. So then you just choose this one. So the base model will match to the variance calling. So you should use the same model that you use to do the base call. Okay? If you use this base call, you use this model, the same model for the variance calling. All right, and then the last step, go to the next flow config. In this next flow config, you have chance to change the name of the run here. Normally, this one gonna be the random name. Yeah, but if you run many workflow, so it's gonna show you as the random name after you finish run. For example, if I show you this, you see? So this gonna be the random run name. And then if you have multiple run, it's gonna show you like many random name, and you don't know what is the sample you're doing. So therefore, I would recommend you change the number or name over here, that you can go back and see which run is which sample. So that means in this step, okay, I need to select it again because I get it back. All right. Okay, can do it really quick. Test, just make it quick. Okay, BAM is there. Reference 210, and then bed file, annotation, then advanced, go to the model that you use, high accurate version 5, and then test 1, -1, or whatever name you want to add. Done. Launch the workflow. So that's all for the running. And now, what happened is that, so the epitome gonna ask the next flow, which is show you over here, the log file here. This log file gonna show what happened during the run. Yeah, and this is what is gonna increase more and more. So this step, what happened is that it's gonna ask the next flow, and this is the next flow version that I install on this computer. And then it's gonna read through the workflow location, right? You have the file name in the workflow, and then it's gonna launch the workflow. And this is all the detail, where's the input information, where's the output information, whereas you are setting here, I have like structural variation true, SNP true, and then copy number variation not show here because I didn't select. So that the option that you provide for the next flow to run. And then it's gonna show you the process here, and if it's die or if it's cancel or stop, you're gonna find what happened over here. So yeah, that's the run. So now I will wait for you to run, and then we will continue to the result. Any problem? It's good? Anyone can run. Yeah, I keep. You to digest a little bit more. So this is shown in the log file. If you go to the overview, this will be show something like the progress bar, which is in the user interface kind of the graphic. But this thing is actually exactly the same to this guy. Yeah, so this is run through command line. All right. So if you have problem, you can raise your hand and then ask help from staff or from me. So I can go and help a little bit. Good? Yeah, just wait a little bit. All right, so good?
Not moving.
Not moving. Oh, so that internet problem. Okay, need to wait a little bit more because some already run and some have the freezing. Here, freezing or not? Launch, yay. Yeah, it's good. It's show running. That's good. If it's show the red color, it's not good. Now it's blue, it's running, it's fine. Is that okay? So, is freezing or? Oh, okay. So if you're freezing, you because you have to select. So right now you need to click on here. Yeah, click, and then go to the home. Go to your data. Yeah, and this is your BAM is over here, so you can open. Click open here. All right. Yeah, that's good. And then you check this one. Same. Click. It's over there, and then go to the data again. Go inside, select the reference. Yeah, and then open. Yeah, the reference here. Then change this one into the 10. Yeah, let's go down. Change this one into the bed file. So go to the data again. And then the genes. Open. Yeah, so this is the bed file. And then next you go to the advanced. Let's go down here. Yeah, advanced. And then select this one, high accurate 5. Yeah, 5.0, sorry. Yeah, and just click again. Go to the high accurate, HAC, yeah. That one, and go down here. Go to the last one, change the name, go up, change the name here, and then you can launch. So I know some have the freezing. Actually, you have to select the window. Yeah. Okay, good. So now is go through now. What happened here? Can you? Okay. So what happened? So it's how it look like. Okay, so it's actually it's launch this one for you, and then you need to select. So what is the bed file, right? Let's go to and select the bed file. It's actually not internet, but it's ask you to select the file. So same to others. Okay, yeah, then go. Yeah. And then number 5, high accurate. Yeah. And then go down, change the name over here. Yeah. All right, so it's installation. Stop with error. Okay, what kind of the error? Okay, so if you have the error, you go to the log here, and the log gonna show you what happened. So what is that? You forgot to select the base call config. So in that case, go back, do again. All right. So you just forgot to select the model when you go to advanced. Yeah. It's running. Okay, good. All right. Okay, install. All right. Home. Go to home. Home. Yeah. Data. Yeah. And what is that? BAM or what? What is the step? Click. You can click. So reference. So go to the reference. .gc. Yeah. Yeah. All right. So you put the BAM already, right? Go up. Yeah, no BAM, so you need to select the BAM and do the same. Click, it's over here. All right, so then go select. Yeah, BAM is over here, then you can open. Yeah, click. You can click open right now. Yeah. Yeah, change the name a little bit. Or you can use whatever name, test, whatever. Okay, so yeah, no, yeah. This one change to 10. So normally keep 20, but this just for this one. Yeah, bed. And select again, go to the data, go to the genes.bed. Yeah. All right, then go to the advanced. Advanced, down, advanced, and then select the model. Yes, yeah. And click, yeah, and then select high accurate 5.0, this one, yeah, And this one done. Let's go down here. Go to the last one. Yeah. And then go up, change the name. Yeah, after you change the name, you can launch. Yeah. You can select this one, structural variation. Yeah, for this one, you can just use these two because the data that I prepare is just two. Yeah, this one you don't have data, so you do not get anything. Yeah, and change the name and then you can submit. All right, so look like most of you are already done. So then we can continue. All right, so I think everyone in the same page now, so just waiting for the run. So you can see that they do many things like do the sequence stat, do the QC for you, and perform the sequencing depth calculation, perform many things, SNP calling, structural variation calling, and so on. Perform the annotation is almost done here. You see, so this normally when you be bioinformatician, so you need to install the software one by one. If you want to study structural variation, so you download the program structural variation and run. If you want to do SNP, you download the aligner, you download variance calling, you download the variance annotation. So there's many software involved into these tools. So therefore, right now today is very, very easy if you can play with or use the EP2Me. So that saves a lot of the time that you have to do. Okay, so now we get back to the slide. All this one we already go through, select the advanced and then select the base core config. And then we change this and launch. So in the overview, you're gonna see the graphic user interface, like the nice progress bar here. You can scroll down and see what is the step is on that time. And then they have the output. So this is the output location on your computer. And then if you want to go to the output, actually you can go a little bit down and then they have the so-called open folder. So then you click on the open folder. It's gonna open the folder of the output for you. And then the output will be in this directory, output directory. So the next one here is the log, as I already show you. So whenever you have problem, so you can go to the log and then scroll down in this area, and you can identify what happened in the pipeline. So just now, one participant have the error in the red color here. So and we scroll down and read what happened, and we can identify that she actually forget to select the base caller model. So then we know what happened. We get back into the workflow again, and then we select all the information that we missing, and then run again. Okay, so that's how you use this log file. And then the result, how's the result look like? Once the result done, so you're gonna have the report over here in the HTML format. So just now it's already finished, but I can check back into my previous run. So you're gonna see in the report here is automatically show you this report, the alignment report. And we actually select the SNP or structural variation, and then we select the structural variation, right? So these two options that we select is show as the report. So you can go one by one if you want to go into the detail. And then this report is actually the overall report of the run. So it's going to tell you how long of the pipeline run on this computer. And for this is 9 minutes, right? And then some other information, where is the output location, what is the CPU used in each step. You can see that this is the all software that you use in this analysis. So it's go into all this process. And they show how much CPU used for each one. And the one that have used a lot is this guy. What is that? Concatenate information. Yeah, so that used CPU a lot. The RAM also show you how much the RAM or memory used during the run, and so on. So that some detailed information. And timeline. So this one gonna show you the timeline of each step of the run. So if you want to track how long of the analysis in each step, you can check on this. Now, the most important is that the alignment result. So the alignment result is going to show you the reads that you use in this analysis. And then show your N50, like the median of the reads, how long of the read here. Total coverage over the reads. So based on our bed file, so it's going to calculate how many of the read cover that bed file. So in this case, we have 14x coverage. And then that's some detailed information about the reads itself. The quality of the reads, 15 or less than 20. So the read length, so most of them is less than 20K. And then, yeah, some accuracy. When calculate accuracy, the accuracy is around maybe 97, 96, because this is the high accurate model, so it's not the super accurate model. So therefore, the accuracy is a little bit lower. And then this is coverage, how much the read cover the region you are interested, and most of them close to the 100, and some of them is about 95%, maybe some missing information on the bed file. All right, so that the alignment to show overall of the reads that map to your region. And then SNP. Okay, SNP information. So it's gonna summarize all the variants that identify the SNP, the indel information. Indel is mean insertion or deletion. That not too long, okay, and show you the TI versus TV. So it's the base transition and the base. So I can't remember the name exactly, so just get back to this slide. Yeah, so the transition. So TI is the transition of the base. When the base change from purine to purine, or pyrimidine to pyrimidine, and the transversion is purine chain to pyrimidine. So this is much more difficult to happen. So this happens when you have damage of the DNA. So, and by default, you will have TI more often than the TV. So therefore, you get this number like 2 up to 3 in the human genome. So if you have this event, TV high, so it means that your sample have a lot of DNA damage. All right? And the most important in that report, in the SNP, is the information of the genes and the variation that cause the pathogenics. So that's the most important. So And we actually, after we run finish, then we go to check the quality, and if quality is good, we go to check this one, whether we have the pathogenic or not. So you can use the search bar here to find only the pathogenic that you want. So, and this case in our sample here, we have 3 SNP present, and only one related to pathogenic, which is BRCA1 or BRCA2 here. This BRCA2 related to deletion of 2 base, G and T, and then make the frame chip, and that means the protein doesn't work. All right, so, and the structural variation, so another things that you can get from the long read sequencing compared to the short read. So long read will cover more the structural variation for you. I will explain more a little bit more later on. So this is the graphical show where the structural variation present in each chromosome. I found that this is not that informative. So if you want to get more information about the structural variation, you can go deep into the IGV itself. That I'm gonna show you in the slide later on. All right, so I will skip this and go into the type of the variation that we do today. So we have so-called single nucleotide variation, the single base different. So if you have chain of the reference into some other base, this called structural variation. So, or some person call about SNP when you use the population information with that single variation SNV. Okay, and indel, when we define the indel, when the indel insertion or deletion is less than 50 base pair. So if you have longer than 50 base pair, those deletion and insertion will classify into structural variation. So, and the list here that you see, tandem duplication, interspersed duplication, inversion, translocation, all of this information related to structural variation. All right. So, and then as we select structural variation, and this cover single nucleotide variation and small indel. And when you select the SV, so it's cover big deletion, big insertion, and all other here. So, and indeed, I already show you that SNP, what happened when we have pathogenic things. So it means that we have change from the normal protein into some weird shape of protein, and it means that it's not function. So if the protein is function properly, it's good. If the change cause the change of the protein, so it means that protein might not work as it should be, or protein might fail to form the structure. It means that you lost the function of that protein. And when we study about the cancer genomics, so you focus on the gene related to DNA repair. For example, if you have the loss of function of these DNA repair genes, so it might cause you the tumor because you cannot repair things in the genome. And also, if you have the mutation or change in the human suppressor gene or oncogene, so you might have the cancer as well. So in that case, epitomy human variation pipeline cover all of these things. Okay, and then I suggest that if you want to see more detail of that information, so use the IGV. So IGV can help you to go deep into the position of the mutation and study or interpret that information using this visualization. So you can go to this link. If you have slide, you can click and then just download. So I have the link to the basic how to use the IGV, so I not show you here. So you can click on the link. yeah, you can click on the link over here and then see at home. So it just quickly show you, this is the example of what we have in our result, the pathogenics one. If we open the IGV and then use the BAM file that you see, the alignment file, you're gonna see this position of the genes. If you can zoom in into that region, you're gonna see the deletion, all right? So this is two deletion G and T, and exactly what epitome report that this is the deletion G and T, and that can cause the pathogenic. If you want to make sure whether this information is confident or not, so you can click over here in the ClinVar, and it's gonna link to another database, so-called ClinVar, that have the evidence for a given mutation to make sure that this is the real pathogenic or not. So, and the last one is the structural variation. Actually, after we run, we get the structural variation located over here, and we're missing this structural variation. You can see this is what kind. So the line here is actually one DNA sequence, and the gap over here that have the line is mean that this one is the deletion, and this the big deletion here actually present because you have multiple reads, multiple DNA fragment that show as the evidence that this is the real one. And you can see that this is the big deletion here that adaptive sampling can capture. But with the software right now, it cannot capture this information. So if you want to find this thing, so you need to go with the IGV and see whether you have big insertion or big deletion like that. So in this case, your data that you have, we have the big deletion of TP53, which is the tumor suppressor. If you lost this function, it means that your cell not protect by the gene grow, and then it can be the cancer. All right, and also this is not in your data, but this is our data in our lab. So we can also identify the tandem duplication. So this one, epitome can identify. So you're gonna have the duplication of one segment 2 times in the data. So that tandem duplication. And also you can identify something like copy number variation when we have loss of the DNA segment. In this one, our sample have loss of chromosome 5, about 5 megabase. So the whole region here is lost in one allele. And it means that you lost all this gene. So one allele. So, and it could be cause of the, or source of the tumor in the future. All right, so that's all. And this is actually, we have the program called Medical Bioinformatics Master Degree in our Siriraj Hospital, Mahidol University. So if you are interested, so it's the international program. So you can submit, and yeah, we will open again next year. All right, you can capture that. Okay, that's all for today. I think the time's up also.
Okay, thank you so much, Dr. Pirun, and let's give a round of applause for Dr. Pirun. Okay, as we come to the end of this insightful workshop, we would like to extend our heartfelt thank you for our workshop facilitator, Dr. Pirun, and also Rebecca, for guiding us through the sessions with clarity and expertise. A special thank you goes to UNDP and BB Genomica for their invaluable support in making this event possible. Their collaborations has been instrumental in bringing together experts and participants from across the globe to share knowledge and foster innovations in genomics. Finally, to all our participants, thank you so much for your time, enthusiasm, and also engagement throughout the workshop. Your dedication to advancing genomic research is what drives these discussions forward. We hope you have gained valuable insights and are inspired to apply these learnings on your own works. We also like to remind you that the certifications for this seminar and workshop will be sent through your email, and there are snacks and also coffee that are provided for you after this, and you can enjoy this afterwards. Therefore, have a great day, and we look forward to seeing you at the future events. Thank you so much.